Okay, sir.
Okay.
Wishing you all a very good afternoon. Live the moments you have at your best, because right after those moments, they'll be best memories and nothing more. Let's all make this day a wonderful, memorable moments. I welcome all the dignitaries for this inaugural function through this online platform. Let's cherish and start this day. Once again, I would like to welcome you all. So not only the dignitaries, but also the participants of these events.
Kurutvam kalyanam arogyam dhana sampadah. Shubham kurutvam kalyanam arogyam dhana sampadah. Shatru buddhi vinashaya deepajyotir namostute. Deepajyotir namostute. Shubham kurutvam kalyanam arogyam dhana sampadah. Shubham kurutvam kalyanam arogyam dhana sampadah.
So I welcome you all for this enlightened lamp lighting. So followed by this lamp lighting ceremony. So we are going to start our day with a prayer. Tamil Thai Valthu. I request you all to stand for the prayer. Thank you, everyone. Valga Tamil, Valartha Bharatam. A warm welcoming always keeps our heart enlightened, gives us joy, and refreshes us with a beautiful fragrance. Here I request our respectable Dean, Professor T.Selvamani, to welcome the gathering.
Good afternoon to everyone present here. Myself, Dr. T.Selvamani, Dean, University College of Engineering, Anna University, Tiruchirappalli. Honored to welcome you all for this inauguration ceremony. MHRD sponsored GIAN Global Initiative for Academic Network virtual course on new concept in tumor biology from the bench to bedside, conducted by the Department of Pharmaceutical Technology, University College of Engineering, Anna University, Tiruchirappalli. I am indeed very happy to welcome all the eminent personalities who presented here for this inaugural session for the FDP. First and foremost, I would like to extend warm welcome to our respected director, Dr. P. Arigaran, Director for Center for Constituent College, Anna University, Chennai, who has supported us for all our endeavors. I welcome you, sir. I am happy to welcome our guest of honor, Professor Martino, Scientific Director, Policlinico, San Martino, Italy, who is here. I thank you, I welcome you, sir. I take this pleasure and I welcoming course faculty, Professor Ulrich Pfeffer, Head Tumor Epigenetic Scientific Director, Polyclinic, San Martino, Italy, who is here with us. Thank you and welcome you, sir, for this your presence in this course. I'm happy to welcome my own team from Anna University, Tiruchirappalli, Dr. Tiruchirappalli, Professor and Head, Department of Pharmaceutical Engineering, and Dr. P.Selvamani, Professor, Department of Pharmaceutical Engineering, and Coordinator for this GIAN course, and all the head of department and faculty members who have supported us for this event. At this juncture, I'd like to appreciate Professor Selvamani bringing all first kind programs in the BIT campus. I'm happy to say he's get the first research from BIT campus, and now he is a first GIAN course coordinator conducted by him. He also helped me a lot when I fight for the legal issue during this compound wall building in our campus, the protecting of 354 acres of land. I wholeheartedly thank Selvamani, gives a lot of support in this junction. I exhibit to welcome all participants and various institutions to this online course. I hope this course will be enriched your knowledge and open to our research perspective in area of tumor biology. Welcome you all once again for this GIAN course. Thank you once again for the opportunity.
Thank you so much, sir. That was a very heartwarming welcome. I thank you once again. Now I request Professor T. Hariharan, Director, Center for Constituent Colleges, Anna University, Chennai, to preside over the inaugural meet. Please, sir.
Thank you. Am I audible?
Yes, sir, you are clear, sir.
Respected Chief Guest of the Day, Professor Tiruchirappalli, Scientific Director, Polyclinic, San Martino, Italy. Respected Guest of Honor, Professor Dr. Ulrich Pfeffer, Head Tumor Epigenetics Unit, San Martino, Italy, and course faculty, Professor B. Senthilkumar, Dean, Anna University, Big Campus, Tiruchirappalli, Professor A. Tiruchirappalli, Head of the Department, Pharmaceutical Technology, Professor P.Selvamani, Course Coordinator, faculty members, staff members, dear participants, and my dear students, good afternoon to everyone. I stand proud in front of you, which gives me a great privilege in delivering the presidential address for the inauguration of MHRD sponsored online GIAN course on new concept in tumor biology from the bench to bedside. Indeed, our youth plays a major role and stand as the backbone of this nation. Nurturing these bright minds will push our nation forward. Such youth march towards success in various fields and departments. Our chief guest, Professor Tiruchirappalli, is a remarkable researcher, assumed office as Scientific Director of Polyclinic, San Martino Hospital, Italy since 2018. As a meticulous academician and researcher, he served in various capacities as Director, Professor, Coordinator, and President of Italian Association of Neuroimmunology. He was a recipient of Vita Levi Montalcini Award, Melvin Jones Fellow Award, Ryan International Research Award. He is ranked as one of the top Italian scientists. Professor Rucci's H index is 55. His research publications are cited 15,847 times by 12,292 different publications. He is a man of great profile and proficiency. Professor Ulrich Pfeffer, Head Tumor Epigenetic Unit, San Martino, Italy, is a renowned clinical researcher, academician who has been always indeed participating and supporting our outreach academic activities since 2016. To see such a supporting personality is not that easy. But today, amidst the busy schedule, Professor Ulrich were with us to grace this occasion. Professor Ulrich will be teaching you all about the very new concepts on tumor biology, what he is practicing currently. Participation in this GIAN course and listening to Professor Ulrich would definitely be a wonderful opportunity for all of us to learn the new concepts in tumor biology. I wholeheartedly welcome Professor Tiruchirappalli and Professor Ulrich Pfeffer for this inaugural session. On the outside, as the Director of Center for Constitutional Colleges, Anna University, I would like to share a few words on this program. Cancer is a leading cause of death worldwide, accounting for nearly 10 million deaths in 2020 and nearly 1/6 deaths. The most common cancers are breast, lung, colon, rectum, and prostate cancers. Around 1/3 of the deaths from cancer are due to tobacco use, high body mass index, alcohol consumption, low fruit and vegetable intake, and lack of physical activity. Cancer-causing infections such as human papillomavirus and hepatitis are responsible for approximately 30% of cancer cases in low and lower middle income countries. Many cancers can be cured if detected early and treated effectively. Scientific discoveries are essential to driving advances in cancer care, from the better screening and diagnostic techniques to innovative new therapies and survey programs are really on impactful research. The main purpose of research is to inform action, to prove a theory, and to contribute to developing knowledge in this field of study. Detailed research enables students to build critical thinking skills, effective analytical research and communication skills, and all these attributes leads to the quality career prospects. Listening to experienced researcher, gaining practical skills are globally sought after and beneficial. Anna University always provides various opportunities to all our students with all these qualities and uplifting them to a brighter and a prosperous future. As one of the constituent colleges of Anna University, the Bharatdas Institute of Technology campus Tiruchirappalli is always been a front runner in teaching, research, and extension activities. The efforts taken by the Dean, Dr.P.Selvamani, and head of the department from the Department of Pharmaceutical Technology are humanizers. Based on the vision and mission of the university, as teachers want our students to bring budding youth to shine in the society, and we are strongly committed in this as confidence flows from competence. This program is sponsored by Ministry of Human Resource and Development, Government of India, under Global Initiative of Academic Network Scheme. At this juncture, they must be thanked for this wonderful initiative to bridge the academic gaps and for the huge financial support. To conclude, I, on behalf of Honorable Vice Chancellor, respected Registrar, and my own behalf, wholeheartedly stand and support in this Human Resource Development Initiative, and once again I thank everybody for giving this golden opportunity in front of this August gathering. I wish all the participants for a pleasant and resourceful learning on new concepts in tumor biology from the learned faculty Professor Ulrich Pfeffer. I appreciate the efforts and support offered by him for this course. I hope you all will enjoy this course and your learning at your convenience via virtual platform. Thank you one and all. Thanks.
Thank you so much, sir. Next. Next, I would like to call upon our respectable Professor Antonio Uccelli, Scientific Director, Ospedale Policlinico San Martino, Italy, to deliver the inaugural address.
Thank you very much for all the distinguished Professor Hariharan, and all the colleagues that gave me the opportunity to be here with you today, also through a virtual connection, and to to build such an interesting program about tumor biology, a topic where the Ospedale Policlinico San Martino has a long standing tradition in terms of science and clinical advancements, and in which many people, such as Professor Ulrich Pfeffer, have contributed substantially through a strong commitment toward high quality science. I think that what you have organized is an extremely important issue within the framework of GIAN, the Global Initiative for Academic Networks, and the role that IANA University has in this initiative is of extreme importance, and we are really delighted and honored that you considered the Policlinico San Martino as a possible partner to build up on science and education for all the young peers that will be the future of the international community working in tumors for the benefit of patients and people of India and all over the world. So really, thank you very much for this opportunity, and I wish to all of you a very fruitful work in the next hours. Thank you.
Thank you so much, sir. So thank you for your time for delivering the inaugural address. Next, I request Professor Ulrich Pfeffer, Head, Tumor Epigenetics Unit, San Martino, Italy, to facilitate the function.
Yes, thank you for this great opportunity for me to share some of my knowledge that I have accumulated in many, many years of scientific research in oncology. And I think it's very important in the world as it is today that international collaboration is enforced. And this is a special opportunity to do that. And I hope that I have seen that the audience is quite diverse, so that we have different scientific and educational backgrounds. And I will try to be at the same time very basic and explicative, but also touch the real important issues that are important at present. So the very advanced aspects of tumor biology. And I hope everybody will have the possibility to follow the course with proficiency. I would like to say immediately that everybody should ask when something is not clear, that also maybe not clear from the scientific point of view, but it may also be everybody of us has an accent in English. I know that I have a heavy German accent, and so sometimes just ask if you don't understand what I have said. No? A special thank to Dr.P.Selvamani, who created the contact and invited me. And I'm looking forward to interact continuously with you. So thank you again. Thanks to the organizers and to all the staff who have made that possible. And yes, I'm ready to start. Thank you.
Thank you so much, sir. Next, I request our ma'am, Professor A. Tiruchirappalli, Head, Department of Pharmaceutical Technology, Anna University, PIT Campus, Trichy, to felicitate over the function.
A very good afternoon to everyone. Respected Professor Dr. P.Selvamani, University College of Engineering, PIT Campus, Anna University, Tiruchirappalli, Distinguished Professor Hariharan, Director, Center for Constituent Colleges, Anna University, Chennai, Professor P.Selvamani, Course Coordinator of this program, our most valued guest, Antonio Zelli, Scientific Director, Ospedale Policlinico, San Marino, Italy, international collaborator of the program, Professor Ulrich Pfeffer, Head Tumor Epigenetic Unit, San Martino, Italy. It's my great privilege to felicitate on this occasion of MHRD sponsored Global Initiative for Academic Network course on new concepts in tumor biology from bench to bedside. As we know well, palliative chemotherapy is given to the cancer patients to increase the survival and maximize the quality of life. Although doctors are still continuing to explore the optimum use of traditional chemotherapeutic agents, the introduction of targeted therapies has significantly broadened the therapeutic options. The recent data suggested that altered pathways underlie the cancer and not just the altered genes. Thus, an effective therapeutic agent will sometimes have to target the downstream parts of signaling pathway or physiological effects rather than the individual genes. In addition, over the past few years, increasing evidence has suggested that solid tumors represents a very heterogeneous group of cells with different susceptibility to cancer therapy. Thus, since therapeutic concepts and pathophysiological understanding are continuously evolving, a combination of current concepts in tumor therapy and tumor biology is needed now. I believe this program will address the underlying concepts. At this moment, I take this opportunity to narrate the legacy of my department. The Department of Pharmaceutical Technology was instituted in the year 1999 with the program of BTech Pharmaceutical Technology. This program is NBA accredited from the year 2018. In 2005, the PG program, M.Pharm Pharmaceutics, and 2007, M.Pharm Pharmaceutical Analysis was introduced. In 2013, MTech Biopharmaceutical Technology was introduced in the department. Later on, in the year 2011 onwards, MS Bio Research and PhD program is available in the department. Around 1,500 students from UG and PG was graduated from the department, and research scholars have been awarded the PhD degree. That is, around 50 scholars have been awarded. Our institute is a front runner of is one of the front runner of the institute in Tamil Nadu and in India. So in our department, so we have established a national facility for drug development for academia and pharmaceutical and allied industries in the year 2010 with the support of DST, Department of Science and Technology, by attracting 6 crores. In the year 2017, another one national facility was established in the department. National facility for bioactive peptides for milk was established with the DST support, with the support of around 4 crores. In addition, we have established a Center for Excellence in Nanobio Translation Research for the research purpose. In addition, the strength of the faculty members, our faculty members have visited various universities at international level by availing their various fellowships. Boycott Fellowship, Masume International Foundation Japan, DST DAD Indo-German project-based personal exchange program, and Africa India Mobility Fund at international level. At national level, they have visited as a visiting professor in various institutes. INSA visiting professor, Tamil Nadu Scientist Award, and Tamil Nadu Young Scientist Fellowship. Similarly, they have been awarded with the various awards. That is, Tamil Nadu Young Scientist Award, Best Innovation Award for their research, and Active Researcher Award. So similarly, many more awards they have been credited. Then in the department, around 7 patents have been awarded, and total number of publications so far published is more than 650 with a good impact factor. So the faculty members have attracted the funding from various funding agencies with a worth of more than 14 crores. Say, for example, UGC and DST, CSIR, INSA, and DBT, ICMR, DRDO. These are the funding agencies in India. So they have attracted more funds from the funding agencies. With the support of various funding agencies, department has well-established, sophisticated analytical instruments under a roof. Our faculty members have made visit to a lot of foreign universities, University of South Florida, University of Copenhagen, Denmark, NUS Singapore, Nottingham University, University of British Columbia in Canada. So they have visited for presenting their research work. So with this, I would like to have a special mention to Professor P.Selvamani. He is the propelling force for taking up this course to all over India and at the global level by networking all major institutions. His dedications and perseverance have always made to step forward in all aspects. I congratulate his team members for the efforts taken to conduct this course in successful manner. So I am very happy to have a guest, that is Professor Pfeffer, Professor Uzeli in this event. Finally, I would like to close my speech by wishing the GIAN program every success. I hope that every one of you will have fruitful and meaningful exchanges in those days. Thank you.
Thank you so much, ma'am. Now we came to the end of our inaugural function. The blessings of God and the wishes of the dignitaries will surely help us to make this program a grand success. Now I call upon Professor P.Selvamani, Course Coordinator, Department of Pharmaceutical Technology, Anna University, BIT Campus, Trichy, to deliver the vote of thanks.
Good morning, Professor Uzeli and Professor Ulrich, and good afternoon, everyone in India. So it has been an honor to me to host this GIAN course on new concepts in tumor biology from the bench to bedside, sponsored by Ministry of Human Resource Development, Government of India. On behalf of the Anna University and the administrators of the university, Honorable Vice Chancellor, Registrar, Director, Dean, Head of the Department, I would like to extend my heartfelt gratitude to our esteemed guest. First, Professor Antonio Uzeli, a Scientific Director, Ospedale Policlinic San Martino, Italy, is a renowned professor and a highly notable researcher in Italy and an extremely busy administrator. It has been our fortune to find his distinguished presence and very inspiring inaugural address. Next, I would like to thank Professor Ulrich, the head tumor epigenetic unit, and this GIAN course faculty from San Martino, Italy, who is always collaborative and supporting to us in most of our endeavors, for his kind presence amidst his schedule and consented to address the gathering and handle the course for the next few days. And my sincere thanks are due to my mentors and well-wishers, Professor Vailraj, Honorable Vice Chancellor, Professor Ravi Kumar, respected Registrar, Professor Hariharan, Distinguished Director, Professor Sivanesan, GIAN Local Coordinator and Director, Center for Technology Development and Transfer, and former CTDT Director, Professor Balasubramanian, the Dean, Professor Sendil Kumar, Professor Purachikudi, Head of the Department, for all their support extended for this GIAN course since its inception, from its application stages till date. Words are not enough to thank their constant guidance and support in the administrative process of this course. I will ever remain grateful to you all, and I thank every one of you for being with us today. I submit my hearty gratitude to all the dignitaries decorated the virtual dais, and I must remark a proficient sense of gratefulness to all our guests for sharing us with some of the finest thoughts which truly inspired all of us. I thank all the directors, heads of the department, faculty colleagues for their encouragement and presence. And I would like to thank the alumni from my lab, Dr. Bibiana, Dr. Sendil Kumar, Dr. Anish Kumar, Mr. Krishnanand, Mr. Satish Kumar, and all other members of my lab and student volunteers from our department who have handled and supported this event throughout till date. And not but not the least, I would like to thank all the participants as well as online viewers for attending this GIAN course. And I wish all the participants a very pleasant learning. And I would like to thank Mr. Shankar and Mr. Balasubramanian, who is coordinating the technical services for this event. And I would like to thank the sponsor, Ministry of Human Resource Development, Government of India. Without their generous support, this course would not have been possible. And I believe, apart from learning, a new network for collaboration in terms of academics and research upon completion of this course to initiate and to undertake tumor biology translational research in India with the support of San Martino, Italy in near future. So finally, I would like to thank who have helped me directly or indirectly in this GIAN course successful. Thank you one and all. Thank you so much.
Thank you, sir. We came to the end of our inaugural function. So once again, I would like to thank all the dignitaries and the participants who are all joined with the virtual platform. So we shall proceed further to the session after a short break. Participants then join in the same ID after a short break. Not
Thank
more
you,
than
Anand.
join in the same ID after a short break.
Yay.
So just tell me when I should start.
I'm leaving you. Thank you very much. I have to
Thank
move back to my other activities, and I wish you all the best for the course. Thanks
Thank
a
you
lot
so
for
much,
giving
sir.
me this. Okay, bye-bye. Thank you.
Thank you, sir. Thank you.
Thank you, Antonio. Bye-bye.
Sir, maybe we shall take another 5 minutes, sir. Is it okay for you?
Okay.
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 Thank you, sir.
Okay. So that would be 1, 2, and 5. Okay.
Yes. Bibiana, once again instructions சொல்லிடுங்க participants-க்கு. So there are few latecomers.
Okay, sir.
And ask them to keep the video on so that we can record your faces. We would like to document your photographs for the GIAN at least sometimes. Thank you.
So I once again welcome all the participants. So general instructions for the participants, the lecture 1 will start in a few more minutes. The participants are kindly requested to rename in the Zoom platform as such in your registration. So some few of your phone numbers have been displayed. So participants are kindly requested to change your Zoom platform name as such in your registration. The first lecture session is on the topic tumor evolution, 2 hits theory and cancer hallmarks. So the session will start in few more minutes. Participants are kindly requested to mute yourself. So you are kindly requested to unmute and disable your video while asking the questions. So if you have any queries, you can ask by unmuting yourself. While unmuting yourself during the query session, so please disable your videos. So please be prepared for that. Participants are kindly requested and cooperated to do this.
Okay.
Yes, I welcome you, sir, for the first lecture of this course. Today we will be having the first lecture on tumor evolution, two-hit theory and cancer hallmarks. So now I hand over the session to Professor Ulrich. So let us listen to him. Welcome, sir.
Okay. And I see you are registering. So I can do you see my slide, tumor evolution?
Yes.
It is important to introduce tumor evolution because it's a little bit the basics of oncology. We have to understand how a tumor forms, how it evolves, and this is by no means simple. And theories have changed over time. We have learned from the detection of oncogenes and tumor suppressor genes, theories on all the processes that are needed in order to form a tumor and to make it grow. And there are very recent developments which have been enabled by next generation sequencing and our possibility today to analyze molecularly many different tissues, many different samples from the same tumor of a patient and from many patients into a depth that was not possible up to a few years ago. And that again has changed our view on tumor evolution. And tumor evolution has obviously consequences for the clinics, for prognosis, for treatment, and for what we can expect tumors to respond to treatments. So, but I have seen that the audience is very heterogeneous. So I try to explain very well even the basics of tumor biology. To start with, what is cancer? Cancer, and I take simply, you will always find in my slides at the bottom where I get the data from, and you can go there and look at it and get more information. So most of the information I'm showing comes from review articles, and in the review articles you will find references to original work, and so you have an access to everything that I'm presenting. So in this case, I'm using the that is available at the www.cancer.gov publications. Cancer is a term for diseases in which abnormal cells divide without control, and that's the central point. Abnormal cells divide without control and can invade nearby tissues. That's the next step. That's a malignant cancer that invades the nearby tissue. Cancer can also spread to other parts of the body, that's metastasis, through the blood and lymph system. Cancer is also called malignancy. We know several main types of cancer. Carcinoma is a cancer that begins in the skin or in tissues that line or cover internal organs. Sarcoma instead is a cancer that begins in bone, cartilage, fat, muscle, blood vessels, or other connective or supportive tissue. Leukemia is a cancer that begins in blood-forming tissue, such as the bone marrow, and causes too many abnormal blood cells to be made. Lymphoma and multiple myeloma are cancers that begin in the cells of the immune system, and central nervous system cancers are cancers that begin in the tissues of the brain and spinal cord. Sometimes the term cancer is used synonymically to tumor, but there are differences because tumor is not necessarily cancer. So tumor is the wider term. Tumor is simply an abnormal mass of tissue that forms when cells grow and divide more than they should, or do not die when they should. Tumor may be benign, so not cancer, or malignant, and then it's cancer. Benign tumors may grow large, but do not spread into or invade nearby tissues or other parts of the body. Malignant tumors can spread into or invade nearby tissues. They can also spread to other parts of the body through the blood and lymph systems, also called neoplasm. So that's the definition of cancer. And obviously the question is, what causes cancer? How does cancer start? And here I would like you to make an input. Are you aware of cancer causes? Can you tell me what can cause cancer? I would like you to intervene, so to say what you know about cancer causes. You surely know cancer causes.
Mutagens.
Pardon me?
Mutagens can cause cancer.
Mutations?
Mutations or the
Viruses.
mutation causing agents like mutagens.
Mutagens? Okay. Hello. So I can write here. Let me see whether I can write. I write mutagens. No, that's not the correct. Yeah, mutagens. Mutagens. Again.
Viruses.
What? Viruses. Other suggestions?
Radiation. UV radiations.
Radiation. So that might be UV is the most frequent radiation you are exposed to, no?
Cancer causing genes which are inherited.
Yes, inherited mutations. Okay.
The use of tobacco.
Tobacco, okay.
Sometimes obesity.
Obesity. So it's diet, let's say generally diet, okay, leading to obesity and leading to metabolic disequilibrium. Well, for example, the World Health Organization also, yeah, diet in general, because also eating too much meat is considered one of the causes of cancer. So, okay, the list is, okay. Other suggestions? Okay, if not, the list certainly is not complete, but it shows one thing, that we are very aware of what we can call environmental causes of cancer. And we also know about genetics. But what we often do not consider, I will show you in the next slide. So this slide shows the causes of cancer, which, as you correctly said, may be environmental or genetic, hereditary. But the real mass of cause, the real biggest cause of cancer development are replicative errors. So simply, when a cell divides, its DNA must be copied. And doing so, there might occur errors. the DNA polymerase has an intrinsic error rate. And that's the main cause of tumors. You see here, in a scale from 0 to 100%, the stronger the red color is, the higher is the percentage of these single factors contributing to different cancers. You see, for example, that in lung cancer, you have a strong contribution, environmental contribution, that is smoking, tobacco smoke, and also for the larynx and head and neck cancers. And you see, for example, melanoma. Melanoma has a strong environmental contribution, which is UV light, and for several other cancers too. But still stronger, for most tumors, the replicative errors are the main cause of cancer. And the genetic risk, there is a genetic risk, but it's certainly below 10% for all the cancers. So what we often underestimate is that cancer is simply a normal consequence of growing cells, because growing cells make errors when they duplicate, and these errors might by chance hit an oncogene and lead to oncogenic mutations that start oncogenesis. This theory has highly debated because Tomasetti and Vogelstein introduced this theory by saying all cancers are caused by a combination of bad luck, the environment, and heredity in this order. So bad luck, replicative errors, is essentially bad luck. And that's a very important point in oncology, because all what happens with the tumor is subject to probability. So it are stochastic processes. The DNA polymerase makes errors, and these errors can occur anywhere. In most of the times, they will not have any consequence. In some instances, the cell will die because an important gene has been hit. And in very, very few instances, a gene may be hit in a way that it determines abnormal growth of the cell. That is what we call cell transformation. A normal cell is transformed to a tumor cell by a mutation. And this mutation is often, or most of the times, is the result of a replicative error when copying DNA. This also means that the more you replicate, the higher your risk. And that means if you have a continuous lesion to a tissue, for example, that's what happens if you expose yourself to too much UV light, or if you smoke, you have a continuous damage to the tissue, and the tissue continuously must regenerate. And regeneration means cell division, cell proliferation. And in this process, you increase your risk that the replicative errors occur, or that by chance a replicative error occurs right in an oncogene. So essentially, we start after fecundation with the fertilized egg. And let's say at that moment, we only have the germline mutations that eventually we inherit from our parents, but we have no somatic mutations. Cancer is mainly an effect of somatic mutations, because as I showed you here, that the hereditary part of the cancer risk is relatively low. That is due, this risk is due to inherited mutations, germline mutations. In evolution, there has not been a real control over cancer causing mutations, because normally cancer occurs in a phase of your life when you are not more reproductive. So the reproductive success is not affected by cancer causing mutations. That's why they are not selected against, especially in populations that do not live so long, that didn't live so long as we do today. And as we will later see, if a mutation in an oncogene, we have no mutations in oncogenes that are in the germline, simply because mutations in the oncogenes are gain-of-function mutations, and they are not compatible with fetal development. We only have mutations in tumor suppressor genes, but we will see that later. So we start with only our germline load of eventual mutations, but we have no somatic mutations. Then immediately during embryonal development, when cells divide, they may make replicative errors. So we have an intrinsic mutation process that starts right after fecundation and lasts for our whole life. After birth, and we are also exposed to environmental and lifestyle exposure, so radiation and all what you said, no, tobacco smoke from our parents, or later on our own smoking, UV exposure, and so on, and diet, no. And we accumulate mutations, replicative errors, and environmentally induced mutations. Among these mutations, at a certain point, there might be a driver mutation, a mutation that is able to transform the cell to form a tumor cell. That leads to the development of a benign tumor. And the benign tumor would not be a problem if it would not at a certain point develop a mutator phenotype. So a gene is hit, it continues to accumulate mutations at a path more rapid than before, because now it divides rapidly. It's a tumor, it divides many, many times, and it divides rapidly, and there might be more replication errors. And eventually, a gene may be hit that leads to even more mutations. So that induces a mutator phenotype in the cell. That leads to an early invasive cancer. And obviously, by doing so, the tumor continues because it's a mutator phenotype. Many mutations accumulate, but not all mutations actually drive tumorigenesis. That's why we distinguish driver mutations and passenger mutations. We say passenger mutations are simple mutations that occur, but that do not influence tumorigenesis. They just are carried on because they occur in the same cell as the driver mutation. And then you have late cancer with many, many mutations. Eventually, that's normally when it's when it's diagnosed and then treated. You start chemotherapy. Chemotherapy on its own induces even more mutations. And eventually, a chemotherapy-resistant tumor will develop, which carries plenty mutations. And that normally is the phase where we cannot anymore stop tumor development. So when we ask what causes cancer, the question can be understood in two ways. What causes the initial transformation of a cell? And I talked about that, replicative errors, radiation, and so on. And on the other hand, what permits the outgrowth of the transformed cell to form a tumor? Because fortunately, not all the initial tumor cells actually form a tumor. The neoplastic transformation of the cell can be seen as the real initiating event of carcinogenesis. Yet, fortunately, most transformed cells do not yield a tumor. Transformation is thus necessary, but not sufficient for the development of a tumor. In 2000, Hanahan and Weinberg published an article on the hallmarks of cancer. Because in order that the transformed cell grows, it must meet certain conditions. It must sustain proliferative signaling. And this often is the starting point. The transformation itself sustains proliferation because it most often hits signaling pathways that control proliferation. But it must also evade growth suppressors that are present in the tumor microenvironment. It must eventually activate invasion and metastasis. Otherwise, it's just a local tumor that can be exported and the patient can be cured just by surgery. It must enable replicative immortality. So because our cells are not able to replicate indefinitely, that's due essentially to chromosome shrinking in replication if there is no telomerase active. So if the telomeres of the chromosomes are not replicated by telomerase activity, and the normal cells, telomerase is not active. So replicative immortality must be enabled. When the tumor grows, it also needs nutrients and oxygen for growing and for continuing to live. And so it must induce angiogenesis. It must induce the development of vessels into the tumor. And it must resist to cell death. That's very important because normally our cells have a system to control for replicative errors and to correct them. And if they are not corrected, a program is activated that we call apoptosis that leads the cell to a programmed cell death. And the tumor cell must block the programmed cell death. These hallmarks in a follow-up article in 2011 have been enlarged by introducing two more emerging hallmarks. That's deregulating cellular energetics, because many evidence has accumulated that the tumor heavily relies on energy availability. That's because most of the tumors show the so-called Warburg effect. That means most tumors are prevalently glycolytic. They just do glycolysis because glycolysis, and they do not exploit the whole energy to form ATP in oxidative phosphorylation. Glycolysis yields much less energy, but that's not the problem. Glycolysis yields pyruvate, and pyruvate is the starting block for all the cellular molecules, nucleotides and proteins that are needed. And so most tumors develop a Warburg effect, that they do anaerobic glycolysis. And this is a deregulation, and that's important. And that may be a target for, on one hand, it may be a target for therapy. On the other hand, it's an environmental condition contributing to tumor development. So especially in people who have a very high energy availability due to a wrong diet, especially sugars, easily to use energy. And obviously, most of the tumors are under control of the immune system. As we will learn in the lesson next week, we have an immune control over tumor development, and the tumor must avoid this control. It must develop mechanisms to escape immune control. We also have learned that tumors show inflammation. They cause inflammation, and the inflammation can be pro-tumoral or anti-tumoral. The tumors that grow and that continue to grow and become invasive and metastatic inflammation is pro-tumoral. And it's a very important point. It's also a point we can act on. And we have learned that at a certain point, there is not just some mutations in the tumors, but we have a general genome instability that leads to many, many mutations, what I before called the mutator phenotype. Excuse me. So when we look at the tumor, that's still work from Hannah and Weinberg, the tumor actually is not formed only of cancer cells. You also have what we call the microenvironment. The microenvironment is formed by immune and inflammatory cells that invade the tumor, and the cancer cells actively attract inflammatory cells, and they produce cytokines that attract inflammatory cells and select inflammatory cells that are pro-tumoral. You have vessels in order for oxygen and nutrient supply, and these vessels are covered with endothelial cells and outside with pericytes that also are different from normal endothelial cells and normal pericytes. So there's a close interaction between tumor cells and all the cells present in the tumor. And at the end, we have cancer-associated fibroblasts. And again, these are different from normal fibroblasts. There's an interaction between those. And eventually, you have invasive cancer cells that leave the primary tumor to invade the surrounding tissue or to enter into the vessels and to be disseminated by the vessels or by lymph vessels to other sites of the body. And you have a primary tumor microenvironment that is different between an invasive and a non-invasive tumor. And again, it's different from the metastatic tumor microenvironment, already simply due to the fact that the metastatic tumor microenvironment is another tissue where other growth factors, cytokines, and so on are present. So what we call the hallmarks, obviously, is all mediated by biological processes in the cell and by specific genes. So the proliferation circuits are mediated by growth factors and their receptors, which most of the time are tyrosine kinases that phosphorylate internal signaling molecules, just like RAS, which is a known oncogene. And that leads to a cascade of events to the activation of changes in gene expression that enable proliferation. And the same is true for hormones, for example, in breast cancer, where you can activate proliferation through hormones. In the invasion capability, you have proteases that act on adjacent cells and degrade the extracellular matrix. And you have integrins, so adhesion molecules, caterines, that adhesion between cells that can start a process that at the end also leads to changes in gene expression. And that is also true for the other facts, differentiation circuits, where you have anti-growth factors that act on cell cycle control molecules and on the central E2F transcription factor that controls cell cycle. And again, you have effects on gene expression. And at the end, you have the death factors, survival factors, and cytokines that induce resistance to cell death. The most known is BCL2, an anti-apoptotic molecule. And you have abnormality sensors in a normal cell that must be inactivated. And the central molecule is P53 because it's a sensor of DNA damage and induces cell death. So that's normally what I told you of many replicative errors. In the absence of P53, many people would develop cancers, and mutations, germline mutations in P53 actually cause a multi-cancer syndrome. So that's the general genome surveillance pathway that is controlled by P53. We will come back to that later. So that's how the hallmark capabilities are activated, essentially by changes in gene expression. This is the older version of the same slide. And here you see that there were always many specific genes indicated in the various processes. But in the meantime, we know so many genes for these processes that it would not be any more possible to indicate all the genes that may act in these pathways. So what is important is that we have these different hallmarks, self-sufficiency and so on. And then, for example, self-sufficiency in growth signals can be obtained by activating the HRAS oncogene through a mutation or through phosphorylation by a mutated tyrosine kinase. You have insensitivity to anti-growth signals. And for example, that might be the loss of a suppressor of cell cycling, like retinoblastoma gene. Retinoblastoma gene is a central regulator of the cell cycle. If this control is lost, the cell does not sense any more anti-growth signals. And evading apoptosis, for example, you can obtain that by producing IGF survival factors, insulin-like growth factor. Or you can mutate BCL2, which controls apoptosis. A limitless replicative potential, and that's normally is obtained by turning on the telomerase, or even by mutation of, that can happen through mutation in the promoter. For example, in melanoma, there's most often a mutation in the promoter of the telomerase. So it's continuously active and the cell can replicate as much as it wants without incurring problems at the end of the chromosomes. Sustained angiogenesis is obtained by producing angiogenic factors like the vascular endothelial growth factor, and tissue invasion and metastasis can be obtained by inactivating adhesion molecules or by producing, for example, proteases that help degrade the extracellular matrix, like the metallomatrix proteases. And what is important, the order of this event is not necessarily only one order. Most often you start with self-sufficiency in growth signals, or with the insensitivity to anti-growth signals. So that's where it can start. But the order of the other mechanisms is not necessarily always the same. You can obtain, it's important that sooner or later in tumor development, you obtain these capabilities. But the order is not important. So, and here we come to an important distinction between oncogenes and tumor suppressor genes. Oncogene, and I take again the definition from the dictionaries, an oncogene is a gene that is mutated, changed, from a form of a gene involved in normal cell growth. Oncogenes may cause the growth of cancer cells. Mutations in genes that become oncogenes can be inherited or caused by being exposed to substances in the environment that cause cancer. So what is important is that oncogenes are normally involved in the control of cell growth. The cancer-promoting function is a gain of function. So the oncogene normally induces proliferation, and if it's mutated, it induces proliferation without control. Normally, the activity of the oncogene is in some way controlled. In the mutated form, it is not any more controlled. And therefore, a mutation of a single allele is sufficient, because if you have an active oncogene in your cell, one copy is sufficient, because it's a gain of function. It has a function that the normal gene does not have. And the opposite is true for the tumor suppressor gene. Tumor suppressor gene is a type of gene that makes a protein called the tumor suppressor protein that helps control cell growth. Mutations, changes in DNA in tumor suppressor genes may lead to cancer, also called anti-oncogene. So the cancer-promoting function is a loss of function, and therefore you need mutation of both alleles. It's a gene, let's say, that blocks cell division, and if you lose one copy, the other copy is still active and it still blocks cell division. Only if you mutate and make non-functionally anymore both copies of the gene, then you get tumor development. Tumor suppressor genes have also been divided in gatekeepers and caretakers. And that means that they function in the control of the cell cycle. So that are the gatekeepers, they decide whether or not to enter into cell cycle and whether to proliferate. And the caretakers that essentially control the integrity of DNA. And the main caretaker is P53. So the cell senses when the DNA is not integral, and it takes care, it repairs the DNA, and it stops proliferation until DNA has been repaired. And tumor suppressor genes, if they are mutated, both alleles are mutated, then this control is lost, and the cell may proliferate even if there are errors in the DNA. So that's a historical slide. So how did we initially identify oncogenes? Essentially what was done, fibroblasts were, in this case mouse fibroblasts, were chemically transformed, so they were treated with cancerogenic agents that we knew that they can transform cells. DNA was isolated and fragmented, and DNA fragments from this cell, from this transformed cell, were transfected into normal mouse fibroblasts, so that each cell receives one piece of DNA. And then you look for those cells that form a clone of transformed cells. You can see that simply by morphology, but also by the degree of proliferation. And then these cells are injected into a mouse, and you see that they actually can form a tumor. And then you can analyze which gene has been received by these cells, which gene has been transfected into these cells. And then you can say this gene transfected is an oncogene. Because again, this is a gain of function. You just inject a copy of a gene in a normal cell and the cell transforms. And this is different for tumor suppressor genes that have been identified by hybrid cells. Tumor cells were fused with normal somatic cells to make hybrid cells. So each cell had chromosomes from both parents, and upon growth, a majority of these hybrid cells did not have the capability of developing tumors within animals. The suppression of tumorigenicity in these hybrid cells prompted researchers to hypothesize that genes within the normal somatic cell had inhibitory actions to stop tumor growth. This initial hypothesis eventually led to the discovery of the first classic tumor suppressor gene by Alfred Knudsen, known as the RB gene, the retinoblastoma gene, which codes for the retinoblastoma tumor suppressor protein. So cell fusion suppresses oncogenic functions, and that hinted at the existence of tumor suppressor genes, which eventually have been isolated then. And in this case, a single copy is sufficient to suppress oncogenesis. But in order to promote oncogenesis, you need mutations of the two genes. So you need to abolish completely the function of the tumor suppressor protein. This is also reflected by the mutations that we find in oncogenes and tumor suppressor genes. Oncogenic mutations are gain of function mutations. And I show you here the cosmic, the collection of somatic mutation in cancer, which is an internet service where you can just write your gene. In this case, I have taken BRAF, which is a known oncogene active mainly in melanoma and sometimes in other cancers, for example, colorectal cancer. And what you see is that it's mainly one substitution, the V600E substitution. So valine is substituted with the glutamic acid. And that's in almost all cases, in 54,923 cases. Essentially, there are other mutations, but they are very, very few. That on a scale of 50,000, you almost don't see them. And they happen into the kinase domain. That's the protein that is produced. This mutation is in the kinase domain, and other mutations are much less frequent in other domains. So that's typical for an oncogene, because you need a specific, in this case, it's an activation of the tyrosine kinase activity. And that cannot be obtained by just any mutation. It can be obtained only by very specific mutations. So since it's a gain of function mutation, so a gene is activated, its enzymatic function is constitutively activated. So you need a specific mutation or very few specific mutations. Oncogenes are characterized by so-called hotspot mutations. Tumor suppressor genes, on the other hand, show loss of function mutations. So the function of the gene must be abolished. I show you here the breast cancer gene 1, one of the major tumor suppressor genes, one of the two major tumor suppressor genes active in breast cancer, I don't know, ovary cancer. And so in order to inactivate the function of a protein, it is sufficient to abolish protein production or to produce truncated proteins. So essentially you can introduce mutations anywhere. For example, if you introduce a frame shift mutation, the frame shift, there's no more functional protein. Or even point mutations with single amino acid substitutions. can abolish the function. You obviously have, in this case, you have some hotspots the same, because even in this case, specific mutations can abolish the function, but you have many mutations throughout the whole coding sequence of the gene. That's typical for tumor suppressor genes. And here you have the protein counterpart. So in order to get that clear, the difference between tumor suppressor genes and oncogenes, if you consider disease severity on this axis and the expression level on this axis, the classical tumor suppressor gene, after the first mutation, it drops to 50% of expression. But still there's no disease. Only if you have 0% of expression, so you have abolished the expression of the normal protein completely, then you have a high tumor risk or disease severity. This is the what normally happens with tumor suppressor genes. But you can also have a haploinsufficiency case for some tumor suppressant genes, where the loss of the first allele determines a medium risk, and the loss of the second allele determines the high risk. And a special case would be dosage model, where the more protein you produce, the lower the risk, and the less protein you produce, the higher the risk. And this is obviously affected by mutation or loss of an allele. And on the other hand, oncogene simply is a continuous model. The more alleles you have, the more expression you have, the higher the risk. Actually, that's according to copy number changes, so amplification of the oncogene. If you consider the mutation itself, the introduction of the mutation dramatically rises the cancer risk. So, but let's come to the real tumor evolution, because as I said, not all transformed cells, fortunately, develop into a tumor, and we have to understand the factors that lead to the development of a tumor. And we have to understand how a tumor evolves during its, and we start from an atypia, so hyperplasia or something in unnormal growth, so that's already a tumor, but it's not cancer. And then we come to an overt primary tumor that eventually invades and disseminates and will create lymph node or distant micrometastasis, and eventually these micrometastases will grow into clinically overt metastasis. And this is obviously dependent on time. So I will show you now a couple of tumor evolution models that are in a way theoretic. And then we will see what the experimental data say. Oh, before I start, that's what we can observe as tumor evolution. So that's a work from Pfeffer and Vogelstein, long time ago, 30 years ago, when they showed that there is a quite ordered process where you go from a normal epithelium to a hyperproliferative epithelium, and then you form early adenoma, intermediate adenoma, and then a late adenoma that takes all the part of the crypt of the intestinal villi. And then you get an invasive carcinoma and eventually metastasis. So that's what we can observe in the clinics. And we have to understand the drivers of these steps and how it evolves. So, for example, that would be the moment where you can detect an adenoma with colonoscopy. And if you do not do preventive colonoscopy, you will detect the carcinoma only when it is already a carcinoma and already invading. And that's why that it makes a big difference when you detect the cancer, makes a big difference on survival. So the colon is a very interesting tissue because you must consider that the inside of the intestine, the lumen, there is passing the nutrients. And in the nutrients, there can be everything. So it's a tissue that is continuously exposed to carcinogens, to toxic substances, to carcinogens, to bacteria, to anything you wish. So it's in evolution, there has developed, evolved in architecture of the intestine that protects the cells from the cancer risk, because you have in the bottom of a crypt, which is actually not exposed to the contents of the lumen, you have the stem cells, the red ones here, that are controlled by the Paneth cells that create a stem cell niche in which these cells are conserved and protected, and they generate the 4+ cells that start heavily. These cells divide only relatively slowly. They generate these cells that divide very rapidly and create all the villi. So that are rapidly transit amplifying cells. And then they differentiate into enterocytes that are the cells that do the nutrient absorption. And together you have enteroendocrine cells that secrete hormones. You have goblet cells that create the mucus of the intestine. And these cells do not proliferate anymore. They eventually die and are released into the lumen of the colon. And this process, these cells are the most exposed to the carcinogenic insult. So the nutrients passing in the lumen of the intestine can damage these cells, but it cannot create cancer because they do not proliferate anymore. Cancer starts from mutations in the stem cells. Okay, that's just to show you where the whole model of tumor development, tumor evolution started by this observation and the knowledge of the architecture of the intestinal villi. So we can imagine a model of linear evolution. You simply start with an ATP formed of certain cells, and by growing, they may make mutations, additional mutations, but they have either they have or they do not have an intrinsic capability to form metastasis, to disseminate and to form metastasis. So either they are not metastatic or they are metastatic. And it's just a linear evolution. The tumor grows. Eventually, if it has metastatic cells, it can disseminate the cells to the lymph node and form micrometastasis, even at a distance. And eventually, these cells grow to a metastasis. This is a linear evolution model. You can have clonal selection. So you have a start with the tumor that at a certain point becomes heterogeneous, and it creates a subpopulation that is metastatic and is selected because only these cells can form metastasis and eventually growing metastasis at a distant point. That's clonal selection. And you could imagine also there is not so much evidence that this actually happens, but I'm quite sure it happens and it should be studied much more. You could have cooperative evolution. You could have different cell populations in a tumor that are able to metastasize to lymph nodes or to distant sites only when both types of cells are there. That requires collective migration. So both types of cells must migrate together into the lymph node and from the lymph node to the distant site. So the interactions of the cells in a heterogeneous tumor are not so much studied, but I'm pretty convinced that some type of cooperation between these cells occurs and that it might be important in the development of metastasis. Also, you could imagine that a tumor grows and has some invasive potential, but not really metastasis potential. But once it has spread to distant tissues, one cell makes a mutation, and this mutation enables the tumor to grow. And at the end, we have the Big Bang model, on which I will talk a little bit more, which is simply that you start with a homogeneous tumor, which at a certain point undergoes genomic instability, creating many, many clones that are molecularly different. And it grows then as a heterogeneous tumor to form metastasis that eventually will grow. The important thing is that for this scope of prognosis, so when what you normally analyze is the primary tumor when it is surgically removed or when you can do a biopsy. You are not able to see the early stages because they don't give symptoms and they are not diagnosed. Normally, a tumor today is diagnosed when it's between 0.5 and 1 centimeter in diameter. Even if you do early detection like mammography, you see relatively small tumors, but still they are some millimeters, no? And they are composed of many millions of cells, no? And at that point, you would like to make a prognosis, which is the risk of the patient to develop metastasis. In the linear evolution model, metastasis is predictable because you can analyze whether it's a tumor that carries cells with a metastatic potential or not. In the Big Bang model, it's also predictable because at the primary tumor, there are already present all the populations that can make a metastasis. If the metastasis-prone cells are not present, then there is no metastasis risk. So you can predict it. Cooperative evolution, if that exists, you could also predict it because you see or you do not see the two or more populations that you need to form a tumor. In the case of metastatic clonal selection, so within the primary tumor, you have one cell or some cells that have a metastatic potential. You could do prognosis only by single cell analysis because the bulk of the tumor is not metastatic and the signal from the few metastatic cells cannot be detected unless you do single cell analysis. And in the case of metastasis-specific mutations, so mutations that develop once the tumor has already spread to other tissues, you couldn't predict the risk because the cell is not present in the primary tumor. And these models that are, let's say, in a certain way, most of them have been observed in human tumors, and some, as the cooperative evolution model, is more prediction, a logical possibility. But what is most likely really the case is that different tumors follow different models of evolution, and several of these models can be combined during tumor evolution. What we know is that for some tumors, let's say, for example, breast cancer, you can predict the risk of metastasis quite well. For other tumors, like for lung cancer, it's much more difficult. And that might also depend on the tumor model that they follow. So I would like to make a short break. Is that okay?
Yes, sir, it's okay. It's okay.
Let's make 10 minutes, okay?
Okay, fine, sir, fine, no problem.
Okay, thank you.
And before closing, sir, let me ask the participants if they want to have something to discuss with Professor. Shankar sir, கொஞ்சம் யாராவது டிஸ்கஸ் பண்றதுக்கு கேட்டாங்கன்னா ஓபன் பண்ணுங்க please. Any participants, would you like to interact? There are few questions in the chat also, sir. What are the triggering factors inducing mutations in oncogenes of TSGs?
The intrinsic factors that introduce mutations. That's essentially, there is a basic error rate in DNA polymerases. So it just happens. They are introducing, I don't remember, I can look for it, the precise, it has been measured. So I think it's 1 in a million. So 1 in a million copies, they simply by chance introduce a wrong nucleotide. Then you have environmental conditions, for example, smoking, the tar contained in cigarettes introduces DNA adducts that then cause the wrong incorporation, the incorporation of a wrong nucleotide. Or for example, UV irradiation forms timing dimers, so that causes a modification of cytosine that is then copied into thymidine. So it's a CT transition induced by UV light. And then you have obviously specific mutations in genes that can lower the control rate. Yeah, that's essentially what we know. On some of these mechanisms, I have later in the presentation.
And one more question now. So the Big Bang model related to stochastic or hierarchical model?
It's a kind of stochastic hierarchical model. Essentially, I come to it. I explain the Big Bang model in more detail later in the talk, so I wouldn't like to anticipate now.
Okay. So the other questions I shall forward you, sir, maybe in the discussion period in the forthcoming sessions. Let the participants interact with you directly. Now, let us break for 10 minutes as you wish.
Yes, you can also write your questions as in the chat.
Yes.
So if you prefer, no, and okay. Then see us again at in 10 minutes.
Okay, sir. Thank you.
Okay, thank you.
Participants, please mute yourself if it is not essential. Since we are recording, we are feeling some disturbance. Please cooperate. Soumya. Yes.
Okay. Okay, everybody here again? So, do you hear me? Everything okay? So I can start to see the questions from the chat. So that are the cancer causes we have. Okay. So triggering factors, I already said, as I said, mainly application errors, but also environmental factors like radiation and so on. We can inhibit phosphorylation of CERN OH group to inactivate. Yes, most therapy is not direct inhibition of the phosphorylation of the serine or tyrosine groups, but to inhibit the kinase. No, so that are analogs of that block the kinase activity, like vemurafenib, the inactivator of BRAF. No, then please tell which amino acid was substituted. In tumor suppressor genes, most mutations are not simple amino acid substitutions, but most often it are frame shifts that cause protein truncation. There are also amino acid substitutions, and there are many. So that's I cannot tell you right now, but if you go to Cosmic and look at BRCA1, you will find some hotspots. And these hotspots, even in tumor suppressor gene, normally are amino acid substitutions, because you can abolish a specific function by substituting an amino acid. But I don't have in mind right now. I can look at it if you wish. Well, why mainly serine? So most of the so you can have mutations that affect directly the catalytic domain of a protein, for example, in oncogenes in the tyrosine kinases. Normally the alterations are in the catalytic domain. And there, obviously, if you have charged amino acid residues, they are a good target, because if you can change the charge, the interaction with the substrate changes. But most of the mutations affect protein folding, so that the folding is different, and then the substrate is no more recognized, or the function is disturbed. No, so it must not necessarily be a chemical, an amino acid that has a side chain that is prone to interact. No, it might simply be, let's say, if you introduce a proline in some position, then normally you have a kink in the protein structure, and the protein doesn't function anymore. That's for tumor suppressor genes, because you abolish protein function. And okay, we will come back to the Big Bang model. I think it's related, all the models that I have to say, I didn't touch the problem of cancer stem cells. So the stochastic model would predict that any tumor cell can grow and is tumorigenic. And the stem cell model predicts that only certain cells in the tumor have the potential of indefinite proliferation, and they create the tumor mass by dividing. This is still not really clear which one of the two is the better model. probably it's different for different tumors, and surely there are in a heterogeneous tumor cells with a different proliferative potential. And I think that the Big Bang model is the best to reconcile the different models, because the Big Bang model predicts a moment of great genomic instability where many different clones are created, and we should expect that there are being created cells that can proliferate and reconstitute the tumor, and cells that may grow but are not really tumorigenic, but they grow together from that point on. So I think that the Big Bang model is good to reconcile many of our theories. Presentation materials, yes, it is being recorded, and I also sent, after each lecture, I sent the lecture, the PowerPoint presentation to Dr.P.Selvamani. Yes, okay. Yeah, and I think the registration will be available. Am I right? Yes. Okay. So we have talked about the different models, and now I step back to the mutations, because all the models rely on mutations. And I wanted to go a little bit more into detail what the mutations can be. So you see, we have many different possibilities, and I distinguish mutations that affect one or few nucleotides and chromosomal mutations. So when you substitute a single nucleotide, this can be synonymous in the sense that the same amino acid is incorporated, and that normally should not have functional consequences. Missense mutations is still a single nucleotide substitution, but missense because a different amino acid is incorporated. You know of the degenerated code of genetic code. So if you hit each amino acid is encoded by 2 to 6 codons, and if the codon doesn't change the amino acid, it's synonymous. If it changes, it's a missense mutation. And missense mutations might or might not have functional consequences. So for example, if you substitute alanine with a valine, it's not so likely that you will have functional consequences, because the charge of the side chain doesn't change. If you change a glutamic acid with a lysine, then the charge changes and you are very likely to have functional consequences. But it also depends on where that amino acid is in the protein structure. Is it in the catalytic center? Is it in a region that is very important for the definition of the structure of the protein? Or is it in just another region where even in missense mutation might not have a functional consequence? And we will talk about that again. Then you have insertions, and insertion can come with a frame shift. So one insertion of one nucleotide or two nucleotide would determine a frame shift. And if you insert 3 nucleotides, you have an insertion that remains in frame. In this case, if you have a frame shift, normally from the frame shift on, you have different amino acids incorporated because the reading frame changes. And so you have amino acids that are not normally incorporated in the protein. And most often you have very rapidly a stop codon just by chance, because the stop codons are frequent. And insertion with frame shift most often define functional consequences, a protein that is truncated and that doesn't work anymore. Or it can also define, with a new stop codon, you may have an instability of the messenger, because there's a control on messenger stability that depends on certain sequences that are normally close to the original stop codon, but they are not close to the new stop codon. That's nonsense-mediated RNA decay. If you have insertion with frame, you insert 3 or multiples of 3 nucleotides. Additional amino acids are incorporated, and it depends on where they are incorporated, whether they can dramatically alter the structure of the protein, or whether they add, they could, for example, hinder the interaction with other proteins because they are right in the interaction interface, but they could also have no effect at all. The same is true for deletions. They might be in frame or out of frame. If you delete 1 or 2 or 4 nucleotides, different amino acids are incorporated, and again, a different sequence, a different reading frame is read, and you have a new stop codon. And in the case of in-frame deletions, you simply have amino acids missing. And again, depending on which amino acids and in which position, this might affect function of the protein. Then we have splice site mutations. That is a nucleotide substitution or an insertion or a deletion, an indel, at a splice site. And we know that splice sites are not very highly defined, so there is no absolute consensus sequence. And after a nucleotide substitution in a splice site, the splice site could still work, but it could also not be, it could also that it is not be that it is no more recognized by splicing factors. And this can lead to exons missing or shortened. So exon missing if the whole exon is skipped because a certain splice site is not recognized. And then the splicing machinery takes the exon downstream. Or it may be shortened when within the exon there is a cryptic splice site that is recognized by the splicing machinery in the absence of the normal splice site, or insertion of unrelated sequences, eventually including stop codons, when a cryptic splice site in an intron is used instead of the normal splice site. And again, it is very hard to predict so if you have a stop codon, if you have unrelated sequencing, you are very likely to have a functional effect. Now, if an exon is missing, this normally affects protein function, or if it's shortened. So normally splice site mutation has an effect on protein function, but it is difficult to predict whether the actual mutation actually abolishes the splice site, because it's not, splicing is not, the consensus is not so highly defined. That is except for the case where you have the first G of the intron or the last G of the exon mutated, then you are pretty sure that it will affect splicing. Stop gain, you might have a missense mutation that creates a stop codon. A new stop codon leads to a shortened protein sequence, or as I said, eventually to nonsense-mediated RNA decay, because the cell recognizes that the new stop codon is much upstream from the old stop codon, but the RNA stability sequences are close to the old stop codon. And you can also lose a stop codon through a missense mutation that abolishes a stop codon. Then you would have incorporation of unrelated amino acids and eventually a new stop codon. Normally, I'm not really aware of cases where this has been seen, but you would expect that the new stop codon is close to the old stop codon, and many proteins might tolerate some unrelated amino acids at the end. We know that because we often work with fusion proteins where we introduce tags, or we introduce fusion to a tracer protein, just like green fluorescent protein, and this is normally tolerated. No, so we expect also that stop loss would be tolerated because just a bunch of unrelated amino acids are incorporated, and the protein function does not suffer. You might also have a loss of the ATG of the start codon through a missense mutation, and in this case you would have no translation, and that would be obviously functionally important, or the usage of a downstream ATG, and that might also have functional consequences. In many cases it will affect protein translation efficiency, because the efficiency depends on the sequence context around the ATG, and if that changes, efficiency drops. And even if you get a protein with the downstream ATG, a part of the protein will be missing, and it depends on the function that that protein has. I'm aware, for example, of a case that's not in the serotonin receptor, where the extracellular domain is abolished by ATG mutation, and the patient was psychotic because the receptor is still produced and still integrates into the membrane, and it still binds the hormone, but in some way the extracellular part that interacts with the hormone binding creates problems. Oops. Then we come to chromosomal mutations. Essentially, you can have copy number gain, so you can have amplification of a chromosome, the chromosomal arm or region of the chromosome. This normally leads to increased expression, but that is not necessarily for all genes in the interval, and there's not a tight correlation to the number of copies. We have analyzed that in some detail or we are right now doing it for uveal melanoma, where amplification of chromosome 8q is often found in tumors that develop metastasis. And for example, on this arm of the chromosome, we have the oncogene c-MYC, which is very well-known oncogene, and initially people thought that amplification of chromosome 8 simply determines metastasis because c-MYC is overexpressed. But actually, c-MYC is amplified, but it's not overexpressed. It's in some way controlled. And there are other genes, and there have been candidates, and we still have not understood which genes are responsible for the effect on metastasis. And that's what we are studying at the moment. And you can obviously have copy number loss, deletion of a chromosome, or a chromosomal arm or region, and that in this case, normally it determines decreased expression. So normally you would have only half of the expression. You have two chromosomes, one is lacking. So you have 0.5-fold expression. But again, not necessary for all genes in the interval, because there might be it might be compensated by an increased expression of the gene from the remaining chromosome. Or essentially, in the case of imprinting, you might also have the condition that one chromosome does not express the gene, and the other parental chromosome expresses the gene. So if you lose the non-expressing chromosome, nothing changes. if you express the expressing chromosome, then the imprinted chromosome, then you will have complete abolition of expression. And then you have inversion of a chromosomal region that can determine the creation of fusion proteins if genes are at the breakpoints of the inversion and they combine in a way that proteins can fuse. So, for example, a gene, you don't need necessarily fusion of the coding region, but you might have fusion in an intron, and then a chimeric protein is generated that contains some exons from one gene and some exon from the other gene. And in many cases, it determines altered expressions, but because the inversion determines a different architecture of transcription factor binding sites and long-range chromatin control. Okay, that are, I hope I didn't forget any type of mutation. So we have many different mutations. And in the case of amino acid substitutions, of missense mutations, I told you that it's difficult to predict the effect. So in the beginning, we simply looked at the degree of conservation of the side chain charge or the side chain dimension, and then guessed whether the substitution might have an effect or not. This has much developed by several different approaches, and I speak just on polyphen 2, which is a method to predict functional effects of human non-synonymous SNPs, and you find that at that internet address. You can just introduce a protein sequence and identify the mutation that you have observed, and then the system will generate a prediction of functional consequences of this substitution. And that is based on a large body of experimentally verified effects of substitutions and a kind of artificial intelligence approach to use the existing knowledge on substitution to predict the effect of new substitutions. And that is important because in addition to the side chain observation, it also contains some knowledge on protein structure, which might change due to a amino acid substitution. A similar system is SIFT, sorting intolerant from tolerant. And you can find that at that address here at that internet site. That are perhaps the most used two systems, but there are many more. Oh, I thought I had a slide. There are many more different sites where you can do that, but that are the two most used. And you can do a single analysis of a single protein sequence, but you can also use the batch tool, where you can insert all the mutations you have found by sequencing approach for the tumors you are analyzing. Okay, mutations occur not always in a completely random fashion. So we have large-scale genome alterations that follow different mechanisms. The first one I would like to introduce is catalysis. That means simply you have a localized hypermutation. So in this diagram, you have each dot is a mutation. And you see that in a certain position on a chromosome, you have many, many mutations of a certain type. In this case, CT transitions. And here you have another example where there are several regions throughout the genome where you have many, many mutations. Kataigis depends on essentially two families of proteins, the 8 activation-induced cytidine deaminase and ApoBec, apolipoprotein B mRNA editing enzyme, catalytic polypeptide. That's a family of proteins that are involved in many processes, among which cancer. And the restriction of retrotransposon, so they are essentially have the function to destroy exogenous RNA and eventually can act on endogenous RNA, and then they create kataigis, so many localized hypermutation. Chromothripsis is another mechanism that is copy number alterations and inversions, and mainly inversions of chromosomes. Essentially, what happens is that a chromosome is chopped up into pieces and the pieces rearrange randomly. So you get large-scale reassembling of fragments with random order and orientation. So here's an example. We have chromosome 14 and you have many, many fusion events where chromosomal pieces have been switched to another portion of the chromosome, and you have also effects on the copy number because some of these are deleted and some are enhanced. And here's a similar example for much more limited event on chromosome 10. Where can you see? So you end up with a chromosome that is has a completely new sequence of fragments contained in the chromosome with many, many effects on many genes. And you can imagine that these two events, kataigis and chromothripsis, normally will lead to a non-functional cell because it's very drastic mutation pattern. But eventually, the one cell that has undergone one of these processes that is still viable will grow. And eventually it will grow faster than the other cells. And that's why it is selected. And that's why we see it in mature cancers. The process of chromothripsis is not so clear yet. It has been proposed that it's linked to micronuclei formation. Micronuclei form when following amplification of chromosomal parts, where the amplified chromosome piece is segregated from the nucleus and exists in a micronucleus with a membrane around it. And this also divides during cell division and might eventually lead to micronuclear pulverization, and sequences of the micronuclear might be reintegrated into the genome, and then you would have the chromothriptic event. A similar thing happens during premature chromosome condensation within micronuclei, where you get, again, micronuclear pulverization that can then be reintroduced into the genome. You have another model is problems during the bridging of chromosomes in in anaphase, where they are pulled apart and chopped up to form many pieces that eventually are reintegrated. And ionizing radiation might also lead to chromosomal pieces that can be reintegrated. In my eyes, the most reliable hypothesis is that we have a case of aborted apoptosis, because in apoptosis, the cutting of chromosomes is one of the steps. And if the cell, at a certain point, when it already started cutting chromosomes in an apoptotic process, is for some way the apoptosis is aborted and the cell recovers, then you would exactly have what you see in chromotherapsis. But this has to be clarified. Still, we do not know. Another mechanism of large-scale genome alteration is microsatellite instability, which has been known for a long time because it was detectable even before the availability of large-scale sequencing methods. No? So we have in the genome microsatellites, which are short repeat sequences. In this case, we show just an A sequence, A polymer. And these are unstable. So that they are less, let's say, they are normally less stable than non-repeated sequences, because polymerases make more errors on repeat sequences than on non-repeat sequences. So they are less stable. That means evolutionarily, normally you see difference in repeat length in different organisms. So between mice and human, you see great differences. And they may become really unstable in tumor cells, where they can shorten or they can become longer. And we call that, and you have tumors where this happens very often on many genes, and they are called microsatellite instability tumors, as opposed to microsatellite stable tumors. No? And you can detect that simply by sequencing of a specific repeat sequence in the tumor, where you see, in this case, a shortened peak and longer peak, and this longer one corresponds to what you observe in normal cells. And this can obviously, if it occurs in coding sequences, it can alter protein function, but most often it occurs in regulatory sequences where it alters gene regulation. And microsatellite instability depends on mutation of specific genes, of a couple of genes, and therefore the tumor, because a specific gene is mutated, the tumor has the characteristic that it will have always unstable microsatellites. And again, in most cases, this determines reduced viability of the tumor cells. And so it's often a positive prognostic factor, but it can also contribute to tumorigenesis if it affects specific genes. And again, eventually specific clones with specifically genes, altered genes, will be selected. So mutations, obviously we have 4 nucleotides that we can mutate, but we have seen that certain carcinogens have different effects depending on the sequence context. And therefore, mainly Alexandrov and colleagues have developed mutational signatures. And here you see an example of that. So below you see the type of transition of the central nucleotide. If you look at the left part where you have single base substitutions, then you see which actual nucleotide transition has occurred. For example, in skin melanoma, you see most of it is red, and you see it's a CT transition. We know that it's due to UV light. And might also come because of other causes in other cancers. Now, CT transitions are frequent, but in melanoma, they are absolutely predominant. You can also see the mutational burden of the single tumors, because each dot here is a single tumor, and you see that, and the scale here is in logarithms of megabases of bases. No, so you have essentially here you have many mutations, and here a few mutations, and you see that melanoma is among the most the tumors that has the highest tumor burden, and then they are ordered in number of mutations. So you have differential mutation burden, and this obviously depends also on the etiology of the tumor. So skin melanoma is always exposed to the radiation, UV radiation, and that determines many mutations. And other cancers are much less exposed, so they have less mutations. But if you consider the sequence context, now I said in melanoma we have a CT transition, but there are 96 possibilities. Let's say you have CCG that mutates to CTG, or you might have ACG that mutates to ATG. So for the single mutation, this obviously makes a difference whether it occurs in a specific site in the protein, where it can determine an altered protein function. But if you look at the genome scale, then you can define mutational signatures, because tumors tend to have specific sequence contexts, and that depends on the carcinogen. So still speaking always on single base substitutions, that would be a signature where you see they are mostly mutations of a C, and CT transitions, but they occur in different sequence contexts. And that are the 96 possibilities now. You have 96 possibilities for these contexts. And when you look what actually occurs in a tumor, we'll see a combination of different sequence contexts that are found in a certain tumor. And that has led to the identification of signatures where specific combinations of mutations are observed. And Alexandrov and colleagues have looked at the frequency and have proposed an etiology that might account for that specific mutation signature. For example, you have, and you see here the frequency indicated by a color code, median mutation per megabase due to signature. So if it's blue, it's very prevalent, this signature in a tumor. If it's yellow, it's not so prevalent. And you can see the different proposed carcinogens. So that's the amination of 5-methylcytosine that maybe come upon different carcinogens. You have ApoBec activity ApoBec, I introduced that as responsible for catharasis, but you also see it at the level of the mutation signature, defective homologous recombination DNA repair and BRCA1/2 mutation, because they are involved in homologous DNA repair, and so on. Ultraviolet light exposure, different signatures that are related with UV exposure, defective DNA mismatch repair, Pol mutation, temozolomide treatment that is mainly observed because you have here, you don't see it, that are different tumors. Now, temozolomide is used for brain tumors, and you see here you have the corresponding signature, which is treatment dependent. Again, ApoBec activity, and so on. Indirect effect of ultraviolet light that you still see in melanoma. So the hope is that we can one day link each mutational signature to a specific etiology and understand better why certain tumors form. And you also should consider, even in melanoma, which is prevalent for CT transitions and the specific UV-related mutational signature, it's not all the cancers. You might also have other causes of the cancer. What is important? The initiating gene, let's say like BRAF or NRAS in melanoma, in cutaneous melanoma, or KRAS or APC1 in colorectal carcinogenesis, the initiating mutation is most often due to a replication error, and it does not correspond to the prevailing mutation signature in that tumor. So BRAF, for example, is most likely not induced by UV radiation. So UV radiation is not really the cause of melanoma. It's a promoting factor. You need a BRAF mutation before, and the BRAF mutation is bad luck. It's an error in replication. And once you have had that bad luck and you have circulating, you have melanocytes in your body that carry BRAF mutation, then you are at risk if you get UV radiation. So that's again the discussion of bad luck and environmental carcinogenesis. And that also explains why people get melanoma after sunburn and other people don't get it. Because it's just by chance. All of tumorigenesis is a probabilistic event. So mutations occur, replication errors occur. They constitute in the first instant a mismatch. And therefore we have DNA damage repair. And before DNA damage repair can occur, the cell cycle is arrested. And if it cannot be repaired, the cell is induced to undergo apoptosis, so to die. That's programmed cell death. And that's mainly the action of a general genome surveyor, which is called P53. It has many other functions. It also is related to senescence or general genomic stability. But essentially in cancer, it induces DNA damage repair. It senses damages in the DNA and induces its repair. It blocks the cell cycle, and eventually it induces apoptosis. That's why p53 is the most often mutated gene in human tumors. Because unless you shut off p53, all the other defects of the cell either will be repaired or the cell will die and no tumor can form. That's why p53 is one of the most important tumor suppressor genes, although it might also have oncogenic function. And you can see that if you look at the mutational profile, that you see some hotspots and many other mutations. And if you go into detail, if you look at the article that I indicate here, you find links to all the functions that are known for p53. And what the impression is that p53 is a very complex protein that has many, many functions. But I caution to see it in this way, because p53 has many functions because it has been so much studied. We have 22,000 genes and more or less 22,000 genes in our genome, and that are far too less to say that each gene has a specific function and just this function. All the proteins have many functions, and we discover time by time. And p53, we have identified many, many functions because it is so highly studied. Similar things occur if you look, for example, at the estrogen receptor. It has so much studied. We know much on the estrogen receptor, but most likely it's not more complicated than any other gene of our body. It's just more studied. But p53 certainly is very, very important because it's so frequently mutated in human cancer. And that's the according protein mutation table. We had that. Oh, okay, that has been remained. Okay. So in different points of my talk, I have addressed driver and passenger mutations, and I wanted to clarify this concept. No, you have a normal cell, and a mutation occurs, and you have a driver mutation. So a mutation that drives the proliferation of the cell, and you form an initial tumor, so hyperplasia. And then you have another mutation, and this mutation may determine more aggressive growth, but it may also not determine aggressive growth. But even if it is not selected, so if it determines aggressive growth, then it is selected, and we can consider it a driver. If it's not selected, because it does not change the tumorigenicity, the proliferation potential of the cell, then simply it grows just like all the other cells grow. But still we find that mutation in the mature tumor when it is diagnosed. So within a tumor, we find many, many, many mutations, very few of which are driver mutations. So mutations that transform the cell into a tumor cell and determine its growth. And they are found almost in all tumor cells because that's the mutations the tumor started with. Bystander or passenger mutations occur in the tumor cell due to its inherent genomic instability and are carried on even if they do not influence tumor growth. And they are not necessarily found in all tumor cells, but it depends on when they occurred and which point of tumor development they occur. And they are most frequent. Essentially, this distinction between driver and passenger mutations is most likely not black and white. So you may have some mutations that really determine tumorigenesis, that drive the growth of a tumor. And you may have, on the other hand, mutations that do not do anything. They are just there. They do not change anything in the tumor. But most likely you have mutations that might have a slight effect on tumorigenesis. And this is a field that has not been studied very much. We have done that on uveal melanoma, which is a good cancer to study this, because uveal melanoma has very few mutations. It's the tumor with the lowest mutational burden. And we have seen that there is an accumulation of mutations in certain pathways, which is significant. So they are in some way selected, but they might not have a large effect on tumorigenesis. That's why you don't find them in many tumors, but you find in many tumors mutations in these pathways that in some way disturb the pathway and help the tumor to grow. So most likely we will have between driver and passenger mutations. We will have secondary drivers or with some effect on tumorigenesis. But generally we adopt this definition, driver mutation and passenger mutations, to distinguish two classes. So the old model of clonal selection predicted simply that you start with the mutation A in a cell, and then the cell forms a tumor. Eventually, there will be a second mutation that confers a growth advantage to the tumor, and then this mutation expands, and again you have a third mutation, and so on. And at the end, you have an expanding cancer clone that carries all these mutations. So that's the standard model of linear of clonal selection. And it's, from the evolutionary standpoint, it's, let's say, similar to what you can observe right now with the evolution of SARS-CoV-2. So yeah, you start with we started with the original variant of the virus, and then it accumulates a mutation that makes it more easily, it makes it more infectious, and then you get a prevalence of the new variant and so on. And this might be true. This is, let's say, that's is the basic aspect of selection, because certainly in any tumor is exposed to selection, because tumors are controlled by the immune system and by many other things, and they must meet all the hallmarks I introduced, and they do that by accumulating mutations, and so the mutations are selected. And the tumor that has, the cell that has all hallmarks fulfilled will be the cell that forms the real tumor that at the end will be diagnosed. And I showed you before the normal evolution of colorectal carcinogenesis, and already in 30 years ago, Pfeffer and Vogelstein identified the likely molecular alterations that determine the steps. So mutation on 5q or familial adenomatous polyposis loss of FAP gene loss define the transition from the normal epithelium to a hyperproliferative epithelium, still a benign condition. Then you observe DNA hypomethylation and you get an early adenoma, still a benign condition, but then you have a mutation in KRAS and you get an intermediate adenoma, which is already cancer, and then you observe loss of 18q to come to a late melanoma, loss of p53 to come to a carcinoma, and then eventually other alterations to form a metastasis. And this model at the time was well in line with this explanation. So you have different mutations and they are selected during tumor growth. But then researchers noted that this sequence is not necessarily always in the same order, and there are many other events that can occur. That is one explanation for one tumor, but not for all tumors. It corresponds to what is clinically observed, and it is important for dissemination, because already an early adenoma starts to disseminate cells. So initially we thought the tumors develop to a large size and then they become metastatic, simply because we thought they do not find enough place in their original location. So they invade the surrounding tumor and then eventually they come to a vessel, to a lymphatic vessel or to a blood vessel, and they invade the vessel and then they disseminate to the whole body. But we have learned that any invasive tumor can release cells. And most tumors do it. And it's not few cells. Let's say a tumor of a small tumor of some millimeter of dimension will release hundreds of thousands of cells each day into the bloodstream. And people do not have metastasis so often because not all the cells released into the bloodstream can make it to form a metastasis, fortunately. Okay. And today we are aiming at detecting circulating cells at a very early step in order to be able to intervene before an overt tumor has formed, and especially before you have metastasis. So, when we study, this is work done by Sotoriva and colleagues, published in Nature Genetics in 2015, and that's essentially the article that launched the term Big Bang tumor evolution model. What they did is they worked on colorectal cancer, and they took a cancer and isolated many, many samples from a single tumor. And they did that for several patients. And they had, especially they had on the left and the right side, and they did copy number analysis, mutation analysis, fluorescence in situ hybridization, methylation analysis to distinguish tumor heterogeneity. What they came up with is that you have certain alterations that you can observe on the left and on the right. And you can observe it in all isolates you have done. And other mutations that are, as this one, present only on the right side of the tumor. So that is heterogeneous. On the other side, you don't find this mutation. And you find still others that are in a very tiny population. And let's say the distance from the origin is time. So you can calculate the time by comparing the different isolates, and then you can say this mutation occurred later than this one. This is the initiating mutation, which you find in all the tumor cells. This is already later, and you find it only in a part of the tumor cells. And then you have private mutations that are only present in small clones of a tumor. And so that led them to the identification of this pattern of time, where you have the different mutations. The starting mutation, which defines clones that contain this mutation. Then the right side specific mutation, which occurred soon after, and other mutations. And that's a completely new model. So if they did it for many different carcinomas, and you can see that it's the rule that you find some public mutations and some private mutations, and differences from the right side and the left side. And you can have tumors with more or less heterogeneity. Here you have only two clones. Here you have more clones and even more clones in this case. But generally, you can identify always the same pattern, that you have certain mutations that have been formed at the beginning of the tumorigenesis that are present in all the tumor parts, and other that are only present in a certain part of the tumor. And that led them to the model of the Big Bang model, because they saw that at a certain point of tumor development, you have the creation of a high diversity. And that means that you have a relatively short time where the tumor becomes genetically unstable and undergoes much mutations. Much mutations means that many of these mutations are not viable, so they determine death of the cell. But some mutations drive tumorigenesis, and from that point on, after genomic instability, there is an outgrowth of clones that contain viable mutations that drive tumorigenesis. And you have development of a heterogeneous tumor with different clones in it that continues to grow. So they predict that the most of the heterogeneity of a tumor is formed early during tumorigenesis, and which is a little different from the clonal selection model. There's still clonal selection, because only clones that can grow and that do grow are selected, but they are not subsequent events. They all are created at the same time point, and eventually afterwards there are additional mutations, but that they occur only in a tiny part of the tumor. So we can summarize this in that way, which we have done in a review article. You have driver mutations that occur early during carcinogenesis. That might be 1, 2, 3, 4, few driver mutations that generate an initial tumor. One of these mutations determines genomic instability, leading to a Big Bang. So the Big Bang, where a high genomic instability is induced. This high genomic instability leads to the formation of many clones, different clones containing different mutations. Because in each tumor cell of the initial tumor, genomic instability determines different mutations. For a certain period, genomic instability is maintained, but eventually clones that remain genomically unstable will die, because sooner or later instability determines also the knockout of essential genes. And what is growing out are clones that are genomically stable and have undergone certain mutations, and they grow out together in a heterogeneous tumor. There's no direct competition between these clones and no selection, therefore. And that's the point where you do diagnosis. You find a heterogeneous tumor with several clones in it. No? And the metastatic dissemination likely occurs already at that phase during the whole process. At diagnosis, either you have already metastasis formed, micrometastasis somewhere, or you will never have, because if you take off the primary tumor, no metastasis will form anymore. And this is obviously on a time scale. So that's in all the time passenger mutations or private mutations occur that might have some effect on tumorigenesis, but normally they don't have a big effect. And if you look at what happens in the mutations in colorectal cancer carcinogenesis, now you have from the start up, you have different pathways, and they are likely determined by mutation either of BRAF or APC. So two different initiating genes that mutate most likely by a replication error and determine whether you develop a hyperplastic polyp or an early adenoma, which then undergoes other driving mutations, KRAS, PI3K, SMAD4, P53, the gene name is TP53, which determines the step from an early adenoma to an intermediate adenoma. And then you have additional alterations, single mutations, single nucleotide variants, insertions, deletions, and loss of heterozygosity. And you have inversions, deletions, amplification, and translocations involving these genes, leading to a full-blown carcinoma. And then eventually you have, again, chromosome instability that leads to chromothripsis in a late stage of the carcinoma. And the tumor heterogeneity is always growing in this procedure. On the other side, you have the first mutation is BRAF, and there's a tendency that you have a mutation of MSH3 and 6 genes or silencing of MLH-A1, and that are the genes together with polymerase E that determine microsatellite instability. And microsatellite instability as a driving force is most likely selected by microsatellites in these 3 genes. And you have a relatively low risk of metastasis in these cancers, because despite the driving force of these genes, the phenotype of microsatellite instability sooner or later determines the non-viability of the cell, especially under treatment. You have MSI phenotype is characterized by slipping frame shift mutations in encoding genes. So that would be the general pathway that you observed, but it occurs in a heterogeneous tumor. But the two ways are separated because the initiating mutation is different. So you had tumor heterogeneity is mostly around the same general theme. So if you have a tumor, it belongs to one class or to the other class, but within this class, there are many different clones in a heterogeneous tumor. I would make another short break, and maybe in the break I can collect your questions.
Yes, sir, sure.
Okay, thank you. Let's make again 10 minutes.
Yes, sir.
Is that okay?
10 or 15 minutes. Take your time, sir.
Okay, thank you.
Thank you, thank you, thank you.
So we can start again if you are ready. Not yet. Okay.
Yes, sir.
So, everybody ready? Can I start again?
Yes, sir. Please, please. Participants, you
Okay,
can also
so-
start your videos so that it can be interactive, as Professor Ulrich told you during the inauguration. Please. Okay. Yes, sir. Please.
Okay, so I see questions. Can a mutation correct another mutation by chance? That's a very good question. In theory, yes, but it's like not so likely. Obviously, a mutation that occurs can also be mutated again. So, but it's not, you have 3 billion of more than 3 billion of nucleotides that you get 2 times the same point mutates is not so easy. So in theory, yes, but in practice, it will never happen. Is there any model that can predict or elucidate the specific type of mutation? For example, frame shift or point mutation with higher rate of incidence in specific age groups? That's a question, good question. Obviously, we have a mutational signature that is related to age, but the mutations that you see in age are an accumulation of replication errors, of simple errors that are not induced by carcinogens. And that signature is seen in almost all cancers because all the cancers occur more in older people. I would have to look at the specific signatures, but you can do so when you go to Alexandrov's work, where you can see which specific mutations are included in the age-related signature. And that, as far as I know, only due to point mutations. Frame shift, I actually don't know. Okay. Are mutations only accumulative in nature? No. So if you intend whether they are cumulative, the effects are accumulative, so additive, or they are synergic, for example. And certainly you have synergic mutations. So let's say you have a mutation in an oncogene that is the initiator mutation, and then you have a mutation in P53 that's certainly synergic, because the only mutation of the oncogene would just determine hyperplasia. And the only mutation of P53 would likely determine, again, a benign tumor. But the two together determine a very aggressive tumor. And that probably occurs many times. So that after the initiating mutation, you have other mutations that are specifically active on that background. That's also because if you look at the mutations you find in a tumor, let's say certain mutations occur in some tumors where they determine an aggressive behavior, but not in other tumors. So you don't see, let's say, RAS mutation in uveal melanoma, whereas you see it often in cutaneous melanoma. And if you test RAS mutation in uveal melanoma, it doesn't work. So mutations are, the effect of the mutations is very context dependent, and it's also very dependent on the mutations that are already there. So mutations are normally not accumulative in nature, but they are synergic, if I understand well the question. So, okay. Why does it not go? Okay. So, okay. I hope you can see now. Okay. So here comes in a certain sense a summary of what we have said so far. That's the EVO index of diversity, and that's change over time, because we have tumors that do not change much over time, that have not a high mutation rate, and other tumors that have a higher mutation rate. And you have more or less heterogeneous tumors, no, very homogeneous D1 or very heterogeneous D2. And if you combine these two things, no, if it's low diversity and low mutation rate, you have a relatively quiescent tumor with a few clones. If even in a low diverse tumor, but you have a high mutation rate, this likely corresponds to recent expansion of a new dominant clone. So you had maybe other clones, but this clone has expanded due to a new mutation. In the case of a heterogeneous tumor and a low mutational burden, you have a neutral evolution with a slow accumulation of different clones, of many clones. But if it's combined to a high mutation rate, then you have a massive clonal turnover. Many, many new clones develop and form a massively heterogeneous tumor. And there are the tumor transitions now. If you have a low diversity tumor with the D1 delta 1 tumor, low diversity, few mutations, it remains either quiescent or it accumulates clones when it develops into a tumor of a higher mutation rate. And the high diversity clones is maybe an old tumor or a neutral tumor evolution. You have a selective sweep to a lower diversity because a single clone outgrows the others. and still with a high mutational burden, or it may develop to a slow turnover if the mutation rate is low. That also says that what we observe as when we speak on tumor models, tumor evolution models, we must expect the coexistence of different pathways in different tumors, but also within a certain tumor type, you might have different portions of the tumor that evolve differently. And during the evolution from the normal cell to the tumor, when it comes to diagnosis and then surgery of the tumor, which is characterized, as we said, by driver mutations that undergo a Darwinian selection. So in the first phase, the tumor is heavily exposed to Darwinian selection. Only the cells that have an advantage grow, and only the mutations that confer an advantage are selected and will be later on found. This, at a certain point when we have the Big Bang, you have the generation of many different clones that all grow fast and determine a neutral evolution of a highly heterogeneous tumor, because you have this phase, the initial phase of Darwinian selection, then you have the Big Bang and the outgrowth of viable clones, and they all have a similar viability. So that really explains tumor heterogeneity as we observe it on mature tumors when they are operated, when they are exported from the body. Another point, talking about the risk of metastasis, that is important. Normally, one cell divides into two, and two cells divide into four, and four cells divide into 8. So we would expect an exponential growth over time. But that's not what we observe. So if tumors would grow like that, everybody who develops a tumor would be dead within a couple of months. What we observe is a quite linear growth. It grows linearly before we diagnose it, and it would grow further if you wouldn't do surgery. This indicates that in our body, there are many mechanisms that can constrain tumor growth. The immune system, angiogenesis, metabolic effects, and so on. And it's important to know that you can do early diagnosis by mammography or colonoscopy or prostate screening programs. Now you can anticipate diagnosis, but still, when you diagnose the tumor, it has several millimeters in diameter, and it has a long history of growth. And it's a heterogeneous tumor. It has undergone all the evolution that it could undergo, and still it's a mature tumor. What is important is that dissemination of tumor cells starts early on. So long before diagnosis, you have dissemination of cells, even if you do early diagnosis. Obviously, you limit the time by early diagnosis that the tumor has to disseminate cells, but still you cannot completely abolish it. And you see that because even very small tumors detected by mammography may develop metastasis later on. And it's also dependent on the let's say on this model, on this model, that the important things have happened early in tumor development. And either it's a bad tumor or it's a good tumor. And depending on how it has been born, it will develop metastasis or it won't. And the evidence for mutations that determine metastasis and occur later on in the tumor development is very thin. I know from several groups who, with the beginning of genomics, have started to compare primary tumors and their metastasis in order to identify metastasis-driving genes, and they had not much success. So essentially, the early development of the tumor decides the risk of forming metastasis. And this is time. This time scale, yeah, okay. So that's the whole time of the tumor development. And that's the time of metastatic dissemination. And you must always consider metastasis is the real problem. Most of the primary tumors can be treated with no local recurrence and with no problem. The real problem are the metastases. Okay. Oops. And still speaking on metastasis and recurring to what I said before, that all of the oncology is probabilistic. In order that a tumor cell can form a metastasis, it must become invasive. It must acquire a phenotype of invasion and migration. So migration must become motile. Invasion means it must digest its way through the extracellular matrix. And that might occur by collective migration. So that's also a little hint at collaborative metastasis models. And it must enter into a vessel, a lymphatic vessel or blood vessel. So that's an active process. The vessel is closed. It must get into it in order to be disseminated. And it must adapt to the metabolic situation and to the blood flow. There's a process called anoikis, which means the inability of normal cells to grow unless they are in a tissue. So most cells that enter the blood flow just die, fortunately. Once it arrives in the tumor, in the metastasis target tissue, it must go out of the vessel. That means it must, first of all, tether to the vessel wall. It must stick to the vessel wall. And then it must actively migrate through this endothelial cells. Then it must seed by perivascular migration, and it must occupy a new niche. So the extracellular, the environment might be very different from the original site of the tumor. That also determines why specific tumors give specific metastatic sites, because if the environments are too different, they cannot grow there. In the target tissue, they are exposed to a massive elimination due to metabolic stress, stromal lethality, and immune surveillance in the new tissue. So they are not compatible with other cells present and or with the growth factors and anti-growth factors released by the other cells, and they cannot thrive there. So they die. but some may enter in dormancy. In dormancy, they can evade the immune system, and they can also evade a growth suppressing signaling. They are just dormant. Then they have to exit from dormancy, where an important factor is MET, c-MET, and they further spread, and acquire mitogenic signaling to develop a macrometastasis, which is exposed to immune suppression. So which must suppress the immune system, becomes resistant to the drug, and often co-opts local cells to grow better. They develop angiogenesis and regenerative trajectories in general, and an immune-mediated pruning. That's the overt metastasis. All of these processes have a probability, and not a very high probability. And that's why I told you before that a small tumor releases hundreds of thousands of cells each day into the blood flow. That determines that at the end, in many patients, no metastases form, fortunately, and in those patients in whom there is the formation of a metastasis, only very few metastases are formed. If this would not be so, everybody would be full of metastasis. So the whole of oncology is probability, and what treatment does, or what treatment can do, is to lower the probability. But the problem is that you cannot target the formation of metastasis because it occurs long before the tumor is diagnosed. You only can target the growth of metastasis, because if you take off the primary tumor, either there are already micrometastases or there will never be, because there's no source of metastatic cells anymore. So you cannot really target the formation of metastasis. You would do it by prevention. But prevention of the formation of metastasis has to be done on healthy patients, because before diagnosis, you do not know that you have a tumor. So essentially, it should be done on all the patients. And I will come to this. I have a lecture also on chemoprevention and on targeted prevention. We will speak a little bit more about this aspect. Okay. Just to summarize what we have learned today, tumors develop from mutated, that means transformed cells. Many other biological processes contribute to tumor development. These processes are called hallmarks. And that's due to the fact that most transformed cells do not grow into a tumor, fortunately. So, for example, we have healthy subjects. We can find, let's say, if you take a skin sample from healthy subjects, we can find BRAF mutations. Or we can even find the Philadelphia chromosome, the translocated chromosome in healthy patients that do not have leukemia. Because fortunately, there are needed many more conditions in order that the tumor grows. The hallmarks, that's the hallmarks that are angiogenesis, immune escape, anti-apoptosis, and so forth. The major cause of tumor initiation are replication errors, and that means bad luck. They occur just by chance. Let's say, I may add, We know relatively little about the genetic background that might affect more or less replication errors. We know that species, let's say turtles, for example, who have a DNA polymerase that makes less errors, they live longer and they have less cancers. So there might even in the human population be differences in the error rate during application. And so there might be not only bad luck, also a part of hereditary cancer. I think that's interesting because the search for, we know, for example, in breast cancer, we know that approximately 10% of the breast cancers occur on a familiar background. But we don't have we didn't find so many breast cancer genes. BRCA1, BRCA2 explain 40% of the hereditary breast cancers. And we have a few other genes that explain another 20%, but there are still 30, 40% where you do not know why in these families occur so many breast cancers. And there are different explanations to this. It could be genes with a low effect, but that work together. So it might be more than one gene. But it might also be, I would say, that among these genes of low penetrance, which have a relatively low effect, might be genes that affect the replication error rate. Okay. Oncogenes contain a gain-of-function mutation. A single muted allele is sufficient for tumor development. Oncogene mutations occur in one or few hotspots where they activate the oncogenic function. Tumor suppressor genes contain loss-of-function mutations. Tumors develop when both alleles are mutated. Tumor suppressor gene mutations can occur anywhere and usually abolish protein function. P53 acts as a surveyor of genome integrity and has features of both oncogenes and tumor suppressor genes. During its development, tumors evolve by acquiring additional mutations. Several tumor models exist. The Big Bang model best predicts the outgrowth of tumor clones after a period of genomic instability. Different models and their combinations might apply to different tumors. And the metastatic potential of a cancer is normally defined early during its evolution, long before diagnosis. Okay, are there questions?
Any questions from the participants, please?
Can you, I don't hear
Yes.
Dr. Pingili. Okay.
Any questions? Anyone?
Okay, if not, I can go on.
Yes.
Because I have not so much clearly distinguished between lecture 1, lecture 2, and the tutorial. Being in online tutorial is always a little difficult, but
Yes.
I have prepared a presentation I have to open just Wait a moment.
Yes.
Let's close that. Okay. Okay, can you see it? Okay. So just 2 words on the analysis of tumor heterogeneity using next generation sequencing. And here I don't know how much you know sequencing technologies, but maybe we don't have so much time. I can go relatively rapidly through the presentation. A brief history of sequencing. Before 1977, sequencing was essentially not available. It was very cumbersome, short stretches, and the sequences were unreliable. Then in 77, 2 different methods were developed, the Maxam-Gilbert chemical sequencing and the Sanger D-deoxy sequencing, which later showed to be much easier and much faster to do. In 96, pyrosequencing was added, which is still a kind of variant of Sanger sequencing, but with another revelation method. Then there was a short time of matrix sequencing where microarrays with that contained all the possible combinations of nucleotide sequences for 8 MERs, so 8 nucleotides, were on a matrix, and you could hybridize your DNA to it, and then with mathematics and in informatics you could single out which is the sequence. This has been brought to application only, as far as I know, to a chip for mitochondrial genomes, because more complex genomes are too difficult to analyze in this way. And in 2005, next generation was introduced, first by the 454 emulsion PCR pyrosequencing method, further on ion torrent, which is still emulsion PCR, but with an electrochemical detection, and then Illumina and similar approaches that use solid phase PCR. In 2016, we had the introduction of nanopore sequencing, and in 2018, the smart sequencing, single molecule real-time sequencing, which are not yet. So at the moment, Illumina solid phase PCR is the most often used method for high throughput sequencing. Okay, sequencing in the beginning was due to the usage of DNA polymerases from E. coli, especially the DNA polymerase 1, where the 5', 3' exonuclease was abolished in order to obtain the cleaner fragment. So you had only synthesis and no control of the sequence. The introduction of next generation sequencing essentially consists in parallel sequencing of hundreds of thousands of molecules and in the sequencing of single molecules. Because before, when we said I have sequenced my DNA, that meant I have sequenced millions of molecules in a single reaction. And so I had no chance to see eventual differences in sequence among these millions of molecules. Whereas now we can really sequence single molecules. How is this done? Emulsion PCR essentially means that you have your DNA, adapters, and DNA fragments, and they are introduced into agarose beads, where in a bead is, you do that by the dilution, in a bead is present a single molecule, and that single molecule is attached to the bead and incorporated in a liposome, in a lipid drop, together with all the reagents that are needed to do PCR. And then you amplify the DNA present on the bead so that you have a bead with many DNAs that are all identical, that are all derived from the initial DNA molecule. And you do this emulsion simply by a machine that produces emulsions of beads and reagents and lipid molecules. The solid phase PCR is another approach where you attach your fragments to be sequenced with adapters to a solid surface in a random fashion. And then you do a PCR on the solid surface so that you come up with islets of identical molecules close to islets of different molecules. So you know that in a certain position, you have only molecules that derive from a common ancestor. And then you sequence directly these molecules. So, and how to parallelize sequencing? So in the bead approach, and that is Roche 454 ion torrent, because both use the same approach up to here. You simply put the beads on a carrier where in each slot, each well, only one bead can enter. And you do that again by controlling your dilution, that you are sure that each slot has a single bead. So you know in this position is a bead with DNA molecules that are derived from a single common ancestor DNA molecule. Each bead carries identical DNA molecules. And then you start sequencing and simply identify the signals that you obtain from specific positions. And in the case of Illumina, where you have on the slide islets with identical DNA molecules that are locally bound, you can sequence directly on the slide, and you will get a signal that derives from one islet or the other. And how to obtain the signal in ion torrent: you have a bead, you incorporate a nucleotide, the nucleotide with the DNA polymerase. The incorporation of a nucleotide releases pyrophosphate and a hydrogen ion, proton, and that determines a change in pH that can be measured by an ion-sensitive metal oxide layer, which transforms the difference in pH in an electronic signal and can tell you whether there has been incorporation of a nucleotide or not. You offer one nucleotide at the time, and you look for which nucleotide is incorporated. And that's the device. In Illumina sequencing, you simply have nucleotides that carry 4 different dyes, and they are incorporated in different rounds of sequencing. And you can, in a single position, see that here it's a blue one incorporated, then a blue one, then a red one, then a green one, then a yellow one, and then again a red one. Now you have the position, and you look in the different cycles which nucleotide is incorporated. And you distinguish the different nucleotides by the fluorophore that is attached to them. Okay? So you see in this case, you have a C, A, T, C, G, T. No? And that's the device. So the sequencing data analysis consists in the acquisition of the raw sequences that come from the machine, the alignment to reference sequences or to the genome, most often to the genome. And if you are working with a genome that has never been sequenced before, you have de novo assembly, which is quite difficult, which you do essentially using the overlaps of the sequences. Then you have a step that filters for quality, because you want to work only with sequences that are of high quality. And you then wish to distinguish between known and new variants, and eventually a quantification. For example, in tumors, quantification is important in order to understand whether a certain variant is present in the whole tumor or only in a part of the cells. The problems with data analysis consist in the dimension of the data, but today most laboratories have organized to be able to store gigabytes and to handle large data sets. You have to do filtering decisions, and filtering decisions are arbitrary in nature. So you decide, for example, a typical filtering decision is to say, when do I believe that a certain mutation is actually there? Because next generation sequencing makes errors, and you have to distinguish between sequencing errors and mutations. But you assume errors to be random and mutations to be non-random. So if you see randomly another nucleotide in a certain position, you say, I don't believe. If you see it many times, then you say, I believe it. And that depends on the depth of sequencing. So how many times do you sequence the same gene? So normally today exome sequencing is run by 100x sequencing. So you get 100, in mean you get 100 sequences for each exon. So you can be pretty sure if you see the mutation 10 times, then you are quite sure that it's not a mutation, sequencing artifact. But you might say 10, you might say 20 to be even sure, you might say 5 to be more sensible. So your filtering affects what you get out. And sequence quality is always a problem. Eventually, nanopore and smart sequencing might overcome these problems. We have many, many applications, and I'm sure in the list I forget some. We have whole genome sequencing, that means sequencing of the whole genome. You do that de novo per species not yet sequenced, or you sequence known genomes for variant detection or mutation detection in cancer. Exome sequencing, sequencing of coding regions of the genome, so sequencing of the exons. Deep sequencing means sequencing with high coverage to detect low abundance variants. So you see, for example, you have it, let's take an example, lung tumors, if they have a mutation in KRAS, if they have a mutation in EGFR, they respond to a certain therapy, to inhibitors of EGFR, and you can detect that by sequencing. And normally you have all of the tumor, most of the tumor carries the mutation. But if eventually the epidermal growth factor receptor mutates again in another position, then it becomes resistant to the treatment. And you would like to know whether the tumor is already resistant. But if it has resistant cells, that are very few cells, but unless you treat it, these cells are not selected because they have no advantage. And you can detect, let's say, a variant that is present in 0.1% of the tumor if you sequence 1,000 times the same sequence. No? Then you can have a high coverage and you can detect low abundance variants. Targeted resequencing has been done to identify variants in a set of genes, and normally targeted and deep sequencing is combined. So in the sense of sequencing few genes, let's say 100 genes, but you sequence them each gene 3,000 times. So you can be sure you can detect even low abundance variants. Paired end sequencing, if you sequence each DNA molecule from long molecules from two sites, you can detect when the two sequences that you obtain from the same molecule are not in the correct position in the genome. And that means that there are structural variants, inversions or translocations. So you get two singles on two different nucleotides, two different chromosomes, then you know there must be a translocation. RNA sequencing for gene expression analysis, simply if you start with an RNA population and transform it into cDNA, and then you sequence it, you can assume that the more often you sequence a gene, the more initial copies were present of the RNA. Chip sequencing, that means chromatin immunoprecipitation followed by sequencing. So chromatin immunoprecipitation has been done before, but now what you precipitate, the chromatin that is precipitated by an antibody against, let's say, a histone or a transcription factor, you pull down also a piece of DNA and you can sequence that DNA by next generation sequencing. ATAC sequencing is accessible transposon for accessible chromatin analysis. Essentially, it's similar to chip sequencing, but uses slightly different strategy. Then we have reduced representation bisulfite sequencing for the analysis of DNA methylation. So you take a part of the genome that is likely to contain methylated sites and you transform it by bisulfite treatment, which determines the incorporation of a T instead of a C. And so, you know, if there's a C, it was methylated. If there's a T, it was not methylated. So you can analyze methylation of DNA by next generation sequencing. And at the end, we have metagenomics, where you sequence not the DNA obtained from just one species, but you sequence all the DNA that you can extract from a biological sample, for example, feces or soil or water. And then you can say, let's say, if you study the bacteria present in the intestine by metagenomics, you can identify with a single sequencing step all the organisms that are present in the intestine. Okay, and I'm sure I have forgot some applications, because essentially next generation sequencing can be applied to everything that can benefit from sequencing, and each day there are being invented new applications. Well, that's the applications I already said. Sequencing starts by the preparation of the template, and then you decide whether to do shotgun sequencing or sequence everything that is therein, or whether you make a selection of regions to be sequencing, like in the case of exome sequencing, or targeted resequencing. Then you do an amplification, or you can do that by PCR or by array enrichment capturing, or solution hybridization capturing, or molecular immersion probes. Most often you use solution hybridization capturing. Then you have a step of DNA fragmentation because you can sequence only small fragments and ligation of adapters and barcodes. So barcodes eventually to distinguish different sources of DNA because you can accumulate different sources of DNA when you have distinguished them with a barcode, and then you do the sequencing. The data analysis starts with either a de novo assembly or a reference mapping to the reference genome. Then you have a filtering, quality control, relative frequency, absolute frequency, germline or somatic mutations, known variants or unknown variants, coding, non-coding region, silent, non-silent, missense, and whatever you want. You do the filtering you need in order to come up with the mutations you are interested in. And then you have the interpretation, which means analyze the effect on the protein, eventually the pathway, distinguish driver from passenger mutations, which you would do by the frequency with which they occur, and eventually with the druggability. So whether there are drugs specific for the protein and maybe specific for that mutation in order to get a clinically relevant information. And for the analysis, there are available many pipelines. I have here indicated the Galaxy server, which Galaxy is a bioinformatic consortium that makes available analytical tools in a way that is also accessible for non-specialists. If you have a bioinformatician in the group, he or she will likely use other resources, not Galaxy, but there are many use bioconductor. We, for example, use R, scripts in R. But all these services have in common that they make available pipelines that map and assemble, call for the variants, annotate the variants, eventually, for example, with polyphane and SIFT, make a functional and structural analysis and an analysis for the pathway level. And then you can come to the hypothesis and generation and testing. The output are FASTA-Q files, so the files with 4 lines per sequence. Line 1 contains information on the sequence that is arbitrary. Line 2 contains the raw sequence letters. Line 3 is again any description you wish, and line 4 encodes the quality values for the sequence in line 2, and you must contain the same number of symbols as letters in the sequence. Obviously, each nucleotide has a code of sequence of quality, a quality code, which is simply the keyboard of your computer, following from the upper left to the lower right, and the higher upper left it is, the higher is the quality. I don't go into detail this. You see that the quality of sequencing rapidly drops. You have so the quality score. Now, if you have a slight quality problem, the quality score drops rapidly. So it's quite severe. When you have your mutations, it might be a known mutation. Then you look whether there are gene-specific drugs available, and eventually mutation-specific drug. And if so, you can propose a therapy. If there are gene-specific drugs, but not there are no mutation-specific drugs available, there might be small molecules available. Then you can check whether these small molecules have effects and propose a trial. If not, you have to abandon this mutation, as you have if the mutation is functionally relevant. If it's not functionally relevant, you are not interested. If it's functionally relevant, you go to the other side, to the search of small molecules. And for these, you have many different informatic tools in the web, for example, cancer and the drug gene interaction database, and that you can, and you have functionally relevant, you do with polyphen or many other similar systems. The effects all are analyzed obviously in vitro assays, and that's an example for the estrogen receptor, which is a heavily studied gene, and its anti-estrogens are in the clinics. You obviously have many drugs for that gene. That's an example of polyphen, where you get for each single mutation an indication whether it is probably damaging or not. So here you have a score of 0.971, so you are pretty sure that this mutation will have an effect. And I would stop here. Is that okay? Have you any questions?
Yes. Any discussion from the participants to the professor?
Okay, probably you are as exhausted as I am.
Yes, I could see you are tired, sir. Anyway, it was very nice for us today to learn many things new. Though we have some information about tumor, but still it is almost refreshing, and we got many new informations from you. And I'm also learning along with other participants from you to improve myself. And let me thank, if there are no questions from the participants, maybe you can write later on also in the chat. Maybe the next day it can be discussed. So
yes,
let me
yes,
thank.
certainly. And there is a question. Maybe there is a question.
Yes.
No, thank you so much. Thank you to the audience, and do not hesitate to collect questions for the next time, or you can also email me. My email is known. Everybody knows my email. I can tell you. I write in the chat. My email is, use this one. Okay.
Yes.
And we see you again next Friday.
Yes, sir.
Same time.
Thank you. Same time, same link. And I
sent you the 2 presentations from today.
Yes. So thank you for your patient listening, dear participants. And thank you so much, sir, for your informative and lecture. It was really exhaustive for everyone, including you, I believe, because we got many informations from you. Thank you so much, sir. Thank you.
Thank you, sir.
Bye-bye.
Bye.
Thank you, sir.
Okay, I leave. Bye-bye.